So, good morning everyone, and thank you for being here. My name is Thesis, and I'm going to present to you my master thesis, which was conducted in the Hellenic Center for Marine Research under the supervision of Dr. Kulur Panagiotou. My thesis title is Integrating Ocean Literacy and Conservation of Marine Coastal Ecosystem, the case study of Elounda Bay. Here you can find a bit the table of contents. Just to let you know, in the bottom right of each slide you can find the number of each slide in which you can refer to for any kind of questions. Okay, starting, we all know that ocean is vital for all life on Earth. It regulates the climate, shapes environments and ecosystems, hosts a large amount of biodiversity, and offers resources, jobs, and minerals to humans. However, coastal ecosystems, due to their proximity to human activities and the great cultural value, the last few decades have been facing increased degradation. To tackle such challenges, global initiatives promote ocean research and ocean education. Ocean literacy, specifically in 2017, has been named by the United Nations as a way to strengthen human and ocean relationship. Ocean literacy is defined as the understanding of the influence that ocean has upon humans and vice versa. Thus, it's important to create an ocean literate society which would serve as a potential ally of future conservation efforts. And that's why we also find it crucial to engage coastal communities and especially the future generation who are going to be the ones facing future environmental impacts. Creating teaching interventions that are site-specific and include the 7 essential principles of ocean literacy, as well as its 10 dimensions such as behavior, attitude, emotions, can assist in the creation of the ocean literate individuals. Thus, we decided to create a teaching intervention based in Elounda, which is located in the northeastern part of Crete. It's an area that has been previously monitored by HCMR. It has an ecological and socio-economic significance due to its highly touristic character. It's very easily accessible by both students and the scientists, and it's an area that's familiar for the students. Some of its basic ecological features is that it's a shallow, semi-enclosed bay. It has an oligotrophic to mesotrophic character. It has a Cauleta prolifera meadow that serves as a nursery ground for commercial species and not only. And also 3 populations of its invasive species have established populations in the bay. These are the Portunus egnis, blue crab, the bivalve Pinctata radiata, and the herbivore fish Gannus luridus. The opportunity came due to the participation of HCMR in the Blue Horizon, Europe funded project called Blue Lights, which promotes blue education across Europe. And it will mark the first ocean literacy activity, which is site-specific in Greece. The main thesis goals are to propose an ocean literacy-based approach for raising awareness to engage students, to assess the ocean literacy level of the participating students and understand common misconceptions that they have about the oceans, and to compare conservation measures of students with scientific proposals for Elounda Bay. The hypotheses are that the students are able to propose such conservation measures, that they are aware of marine topics and anthropogenic pressures of the oceans, and that the teaching intervention is going to affect their ocean literacy level. To collect scientific and scientific measures for Elounda and measures from the students, a mixed method approach was followed. First, a literature review gathered proposals from the monitoring reports and academic databases, and then to assess the impact of the teaching intervention as well as the ocean literacy level of the students, we conducted ocean literacy surveys which had the following sections and were distributed before and after the teaching intervention. The ocean literacy surveys were differentiated for the primary school, which was the elementary school, and the secondary schools, which were the middle school and the high school. Finally, the teaching intervention was organized in 2 different days for each of the schools. The first one was conducted in the HCMR building, where students attended a presentation related to Elounda's environment, and then lab sessions about the blue crab and sorting and identification of marine organisms. And the second day was conducted in Elounda, where students had the chance to engage with local stakeholders and also participate in a stakeholder simulation game in which they had the chance to propose their own conservation proposals for the protection of Elounda. The majority of data analysis was conducted using our studio. For the survey's responses analysis, we used mainly non-parametric tests, depending on the type of data. For the internal consistency of the survey, we calculated the Cronbach's alpha, and while pre- and post-intervention comparisons took place, and school-based differences were investigated to understand better the impact of the teaching intervention across each section of the survey. Lastly, we followed the thematic analysis to recognize general categories of the conservation proposals of the students and also calculate the relative frequencies. Moving on to our results, the literature review highlighted the importance to conserve the marine environment of Elounda and its ecological significance as well. Among the measures that were mentioned was first to limit the boat speed, to prevent segment resuspension, which is causing damage to the Kleptoplasty prolifera meadow, to create targeted fishing for the Siganus luridus, which was observed to feed on algae in general and also Kleptoplasty, and for the Portunus segens, which was observed to feed on juvenile fish inside the bay, to create a sustainable fishing regime for the commercial species of the bay, which have been facing decrease due to overfishing, a long-term monitoring of the invasive species, of the general function of Kleptoplasty prolifera meadow, and also the pollution sites that have been found to be inside the bay. Creation of a sustainable aquaculture based on the local bivalve Arcanoe, which used to be a commercial species for the area, and it's used as a bioindicator for pollution. Coastal erosion measures prevention, and lastly, raising awareness among locals and tourists using virtual tours and the creation of stakeholders dialogue for the protection of the bay. Now, when we analyzed the results of the survey, we found out that actually the majority of the students, independently of their age or school actually, had previously participated in a kind of environmental activity in the past, which was interesting. They also showed that the sources that they mostly used to take information related to marine ocean issues across all the schools were mainly TV and internet, while for elementary school students, they were heavily dependent also in interpersonal sources, such as school or family and friends. And in terms of the most trusted sources that they had, schools was rated the first in both pre- and post-intervention, while after the intervention there was increasing trust for museums, books, and TV, which indicated a positive shift of awareness after the intervention. In terms of the internal consistency, the Cronbach's alpha values revealed a poor consistency, specifically in primary school survey across all sections, and that was attributed mainly to the generally low ocean literacy level of the elementary school students, due either to their young of age or to their limited exposure to marine issues. While for the secondary school survey, the Cronbach's alpha was low only for the knowledge section, and that was more explained by the fact that the questionnaire was formed by a mix of differently difficult questions, or the content of the questions were mixed as well, creating long-term consistency, since students do not conceive all types of the principles of ocean literacy in the same way. Some are easier than the others. So the various sections that were analyzed, first was the knowledge, and we saw that both secondary schools, middle school and high school, had a high knowledge level after the teaching intervention, while for the elementary school it remained stable. And also both secondary schools, both after and before the teaching intervention, at a higher ocean literacy level than the elementary school students. Some of the main misconceptions involve these three principles: that the Earth is one big ocean with many futures, that the ocean supports a great diversity of life and ecosystems, and that the ocean and humans are inextricably interconnected, while also an increase in the correct answer is related to the marine environment of Elounda after the intervention was also observed. For the perceptions and behavior sections, it was observed that there was no kind of difference after the intervention. However, their mean values indicated a positive leaning, stand, and ideology. In these sections, the main questions that had low mean values involved complex marine issues, such as coastal erosion and climate change. The experience and emotion sections revealed the connection that students had with the sea. So firstly, we saw that as an experience, we saw which activities students prefer to do when they go to the sea, and both before and after, swimming and snorkeling were the most popular ones. While in terms of the emotions, they mostly felt positive feelings such as happiness, excitement, serenity, and these feelings were explained by words such as relax, fun, swimming, waves. And their negative emotions, which included mainly fear and anxiety, were explained by the fear of sharks, trust, and the fear of the unknown. In terms of the future, but also of the unknown of the sea. Okay. So, lastly, the thematic analysis revealed that the students proposed a diverse set of measures that mostly had to do with the regulation of the tourism sector, the lifestyle changes, and fishing regulations. Some examples can be found here. What was interesting was that we saw that they didn't propose as many measures related to scientists as we would expect, and that was attributed to the fact that they didn't understand exactly how scientists contribute to conservation of marine environment. However, for other categories, such as fishing regulations, we saw that they had more relevant measures to the ones that we proposed, that the scientific community proposed, sorry. So, moving on to the final part of this presentation, this project confirmed the feasibility and value of involving students in conservation efforts, especially when they are grounded in a local context. Not only it provided proof of the ecological significance of Elounda and showcased the lack of implementation that has led to its degradation, but also created a guide of conservation measures for future management purposes. In terms of the students' ocean literacy level and the impact of the intervention, students at the moderate, even students at the moderate ocean literacy level, we saw that their knowledge was increased in most of the cases. However, for elementary school students, it's proposed to use a more age-adapted approach and a more practical one, so that we can increase their engagement. The recognized misconceptions were mostly attributed to the fact that in the Greek school curricula, there is largely missing the topic of marine science in general, or the fact that some marine concepts are quite complex for them to comprehend, such as climate change, and they do not fully understand their impact on their life. The stability in the perceptions and behavior section was explained by the requirement of a longer and more continuous approach, so that we can create so that we can spark change for them, because behavior and ideology are two sections that are quite difficult to change for students. And however, experiencing emotions and their strong connection to the sea highlighted potential stewardship for future conservation efforts, since they feel so positively about the sea. In terms of their proposals, we saw that they were able to provide relevant measures to the ones that scientists proposed. However, in some cases, there was lack of connection with ecology of the marine environment of Elounda. Such examples were like placing recycling bins or riding a bike, which also explained the low percentage in other categories. However, we saw that in fishing regulations, they were able to propose measures that were heavily interconnected to the information that we previously provided to them. So we understood that ocean literacy can actually be a bridge between community and scientists, and help in closing the gap between them. And also can be used in other coastal marine areas or marine protected areas for similar purposes. Some limitation was the short-term and theoretical approach that we had for this intervention, as well its broad character, which led to a very long survey. So for the future, we would recommend a more specific intervention that is based on a student-selected topic and combines theory and experiment together to create a bigger impact. Closing, school curricular largely missing, as I said before, of marine topics, and this has led to 5 different ways to empower students in marine conservation. This study provides an optimistic example and guide of how ocean literacy site-specific interventions can be organized and work, especially in Greece, to be honest. And it has also followed criteria for EU Blue School Network, such as engaging engaging students of different ages, involving the local communities, and exploring the topic from an interdisciplinary way. The Blue Schools are a network of schools that promote blue education by promoting ocean issues as projects inside and outside of the school. And it's important to keep in mind for the future that such interventions should be financially supported synergies of different stakeholders and educators and scientists to create a long-term intervention with a big impact on students that will be, at the end of the day, the future citizens that are going to have to make informed and responsible decisions about the marine environment. Interventions like this can be also used in marine protected areas. Here you can also see some of my conclusions. And at this point, I would like to thank my supervisor, Dr. Elounda, Athanasio Moya for the help with the manuscript and the data analysis, Eleni Kubaki, Eleni Psonki, Maria Maidanou for their help with the teaching intervention, as well as my family and friends who supported me through this experience. Thank you.
Maybe now the question from the evaluators.
Yeah, I can start. First of all, congratulations for this very nice presentation, which I really enjoyed. I'm definitely not a specialist of ocean literacy, but really enjoyed that. Maybe my first question was about something you said in your very last slide. You said that this work, how ocean literacy can be used in power conservation efforts, and then you had, especially in Greece, to be honest. Why especially in Greece?
Now, I think it can be used in a European or non-European context as well, but I think that in Greece, due to the fact that we are largely missing such topics from schools, I think it would be very interesting for students that have already a connection to some coastal areas to base such interventions in a local context, because students have already, as we've seen in the connection part of the results, they have a connection with the sea since they mostly spend all of the summers there. So creating approaches with areas that students feel connected to, it's more likely that it's going to create an impact for them as well.
Okay. Maybe another question about the choice of the public. So you had about 30 students per age class.
Yes.
How was it chosen?
So the thing is that the Blue Lights project gave us the opportunity to have some schools that participated in its experimental phase. However, there are some schools that already have a connection with HCMR, since HCMR is creating such activities already in the past. So these 3 schools actually reached out to us and they said that they would be interested to participate in a kind of teaching intervention, let's say. They proposed that they wanted to talk about the elementary school, if I remember correctly, was interested in an archaeological site, and Elounda has also a very large history because it has Byzantine ruins. It also was connected to ancient Greece, so it has also historical value. The middle school was a blue school actually, so they had already in the past participated in similar experiences and they were interested to explore Elounda because they were from a very close by area. And the high school actually was a music school that was very interested in understanding anthropogenic pressures on a marine environment, so we recommended to them to follow this kind of teaching intervention and understand better how humans are creating pressures from marine environments. So that was the way.
Yes, you will someone or
Maybe you will.
yes.
Yeah.
Okay. Congratulations, Sally.
Thank
You
you.
speak Greek?
Yes, of course.
Yeah.
I'm from Greece, sorry.
For the surveys, it's important
Yeah.
for us to establish the connection with the target. No? A few questions.
Of course.
First is congratulations for your work, nice presentation. The second is you were talking about the Chromback
Yes.
index, and this is about the relativity no? of your. But what about the validity? Did you test the items in some groups with a differential type of education or something to validate your items or not? It's just the Chromback, the alpha Chromback.
So I did the Cronbach's alpha to understand whether each section was having showcasing the same character, let's say across knowledge, perceptions, and behavior mainly, because they had the numerical data. And then for the knowledge section, I also did personal item maps, which I didn't include in the presentation, just to understand a bit the difficulty of each question across the schools. However, I tested for each section separately the differences, but there was no significant differences in many of the sections.
No, my question was that it was recommended to test the items previously with a spare or with a group with differential,
no.
just to check the validity. Because one thing in surveys is the reliability, and another thing is the validity of the information you're using. And this is just an advice because it's difficult to publish this because we are marine scientists, and then you have to face with reviewers that are experts in surveys, etc., and they ask both things. It's just a recommendation. Another thing is, after were you worried? Because, I mean, the results about the perceptions, emotions of the student is low, no? Is in the program, the project in Blue Lights, there are the intentions to try to modify the curricula and try to introduce, I mean, the project has that as a target or not?
So the Blue Lights itself, it's something separate, I think, in terms of, it provided the framework and the opportunity, but the teaching intervention that we organized was a bit separate from that. So Blue Lights wants to just promote it. They do not, it's important for them to create an impact and to leave something behind, and especially for educators, to have like a platform where they can refer to. However, for the specific teaching intervention, they were not exactly in participation 100% with, but we used the opportunity to also promote ocean issues. Can you repeat again whether I was worried in terms of the low
level of
I think
connection with the sea in an island?
I think I'm more worried with the fact that it already exists rather than the fact that this opportunity didn't have like the huge impact that we would wish for. I think that I'm more worried with the fact that Greek students that are surrounded by water all day and all, they have no idea about basic concepts about marine environment, rather than the fact that, for example, what I organized in 2 days, in totally 4 hours, didn't have a huge impact. I think, how I can approach it next time, and I'm very happy for recommendations also, and I'm very passionate about this, so I really want to be able to create something that each student will be able to say, okay, yes, today I learned this, for example, for the sea, and I think I would be very happy with that, actually.
Thank you so much.
Thank you for your presentation. I really enjoyed it, and I'm wondering how you prepare the questionnaire and how you propose the questionnaire to different kind of people. So it was the same questionnaire, or you modify it according to the public. I mean, for primary and
secondary
school, they are not.
Yeah. So how I prepared for it? was quite interesting. I think like we tried to find, my supervisor recommended some questionnaires that have been previously used, and then I did a thorough research in terms of other questionnaires. There is a questionnaire proposed by Favil in IOLS, which was a big ocean literacy survey that was conducted some years ago, and they have like many different questions related to ocean literacy evaluation, let's say for students. And so we collected many different questions, and we saw which ones were more suited, let's say, for our teaching intervention. And also we added questions about the Elounda, because we wanted to also understand whether students had any kind of knowledge about the Elounda before. This was the main part. Then we also considered the dimensions, as I mentioned in the beginning. We wanted to be sure that most of the dimensions actually were going to be present at least in one or two questions. So we had to also create that. And then we had to adjust the language for both secondary schools and the primary schools and remove some questions from the primary school. I think it was a long kind of a discussion between me and my supervisor, and also some people that came in to help in terms of how we can make this questionnaire work better for schools. And we also changed slightly the language of the for the elementary school students. It actually needs to be even more changed and be even smaller as a questionnaire, because we saw that actually students struggle a lot, even in secondary school, to follow long sentences, fully understand what they mean. They get confused with like small words that change. So yeah, I think it's very interesting to think about it, like to find creative ways to evaluate ocean literacy in such cases. I hope I covered the...
Yes, yes, perfect. Thank you. And another, maybe it was a curiosity.
Yeah.
What about, you talk that you start with a literature review.
Yeah.
So do you find any difficulties in finding some information in this kind of literature review? It was easy or not?
Okay, so that was another journey, let's say. In the beginning, I went very much broad with this. So instead of like looking articles specifically for Elounda, I started searching for articles for each of its ecological features. That was a very painful experience, I would have to say. It was very long days in front of a computer, and I quickly realized that I have to isolate specific information. However, later, I specifically focused in Elounda. In Web of Science and Scopus, it was easy to find the information that I needed because they were limited articles. In Google Scholar, because I also searched that there, huge amounts of grey literature or articles that were more related to geology or historic things about Elounda. So at the end, there were around 20 articles more or less, but I also searched in the previously done, let's say, literature review for specific measures that were proposed in similar areas. For example, there's another area in Spain that has similar features, like it has a Kleptoplasty prolifera meadow as well. It's very touristic. So we also took some information from there to compose in the manuscript. Here we mainly propose the most big, let's say, ideas that were found in literature.
Thank you. So I think we will start with the question and we will go to the next. So something I forgot to say, if I stand up, means that we have to wrap up. So now we will stand up 1 minute before the end.
Okay,
So that you know that.
that was good.
Can you all hear me? I'm speaking loud enough? Okay, great. All right then. I'm Karis, and welcome to my thesis defense, which is on the topic of living boulders and eco-engineering with wetlands to enhance intertidal biodiversity. So to start off, we'll go through the introduction as well as a bit of context on the project before the aims and hypotheses, materials and methods, the main results, the discussion of the results in the context of literature, and my final concluding remarks. So to start off with, urbanisation is increasing all across the world, and unfortunately, as can be seen in this map, it is not limited just to land. It's also extending into the sea, with hard engineering structures such as coastal defences, maritime infrastructure, and recreational infrastructure expanding into our oceans in a phenomenon known as ocean sprawl. Unfortunately, the impact of this habitat loss and this habitat replacement, which is primarily made out of vertical, homogeneous features and differing in material infrastructure, means that we lose a lot of important microhabitats, which of course has negative implications on ecology, specifically in relation to reduced protective spaces and increased spatial competition. Eco-engineering, or the redesign of ecological engineering infrastructure to facilitate ecological co-benefits, can enhance natural features that are lost, such as water-retaining features, material cues, and textural complexity. Previous solutions have been done on things such as seawalls, such as retrofitting complexity tiles or integrating flower pots to retain water, which enhances these features that are lost through the changing of the coastline. Riprap revetments, or large pieces of boulder which are placed along a sea line and a seashore to reduce the impact of coastal erosion, may have greater complexity in terms of the niches that are available in the areas surrounding them, as well as the areas in between the boulders. However, one key natural rocky shore feature is missing, namely rock pools which retain water. So one research gap that exists is whether the integration of water-retaining features into riprap revetment design can enhance biodiversity. And one potential solution for that is that of the living boulders. And in this inaugural pilot study, eco-cement boulders were created with 6 different study rock pools in them. And these rock pools have got 3 different textures, namely smooth, honeycomb-pitted, and horizontally ridged, with honeycomb-pitted and horizontally ridged mimicking textures found in natural rock pools. And these modules can be placed into the riprap design to enhance water retention. So, for my aims and hypotheses, I was looking at whether living boulders can enhance biodiversity on riprap revetments at 3 different scales. First of all, at the site scale, so whether the presence of living boulders in a revetment initiates spillover effects across the entire revetment. At the boulder scale, so whether biodiversity differs locally on the living boulders when compared to revetment boulders that are pre-existing. And at the microhabitat scale, so whether increased textural complexity in the rock pools enhances biodiversity, and whether these artificial rock pools are able to mimic the natural assemblages found in natural rock pools. And this was done for both sessile and motile biota, benthos, and for fish at the site scale. So, going into my sampling site, the study was conducted in Lavender Bay, in the Sydney Bay Harbour, just beneath the Sydney Harbour Bridge, and it was deployed in April of 2023. In total, 5 different sites were screened. First of all, a modified revetment site, which is the riprap revetment with the living boulders integrated into it, as well as 2 controlled revetments, which are stretches of the same riprap revetment but without the living boulders. Additionally, 2 natural rocky shore reference sites were used in being able to compare natural rock pools. So, going into the study design, specifically the site and boulder scale. First of all, 15 living boulders were deployed at high, mid and low 5 of each, and these boulders have got the study rock pools that I mentioned, which means you're able to investigate the influence of water retention. However, because they're also made out of this eco-concrete, we have to be able to control the influence of material. So an additional 3 control boulders, which are the same material but without these study rock pools, were deployed at mid-tide height. Additionally, 15 pre-existing sandstone boulders were measured per revetment site. So to go through the study design slightly, at the site scale, this consisted of comparing sandstone boulders at 4 different time points across the revetment sites, one of which the time points was prior to installation to see whether we could see a divergence into community composition with time with the installation of the living boulders. Additionally, at the boulder scale, this was done just at the modified site and was the comparison of sandstone and living boulders at the 3 tidal heights mentioned before. And for the material effects we investigated, this was sandstone, control, and living boulders just at the mid-tidal height, since that's where the control boulders were. Now at the microhabitat scale, 15 living boulder rock pools were investigated, 5 of each of the different textures mentioned before, all of which were post-installation, of course. And to investigate how well biomimicry was happening, 5 natural rock pools were measured per reference site and compared against 5 of the smooth living boulder rock pools in our study location. So, for the sampling design, first of all, for bedrocks, in situ quadrat sampling was done, and for the boulders, this consisted of the tops of the boulders, so being able to investigate the water retention devices, as well as the sides of the boulders, which don't have any water retention devices. And within the rock pools, micro-quadrats were done in the basins themselves. For motile, this consisted of census counts, whilst for sessile, this was point-intercept estimates of coverage. So, and for fish assemblages, this was done just at 22 months as a primary study, and it was done over 3 consecutive days with 3 replicants per site, and unbaited GoPros were deployed at high tide with 30 minutes of video footage analysed per rep. For the statistical analyses, I'm not going to go into that in too much detail, so if you have questions at the end, feel free to ask. However, alpha, beta, and functional diversity indices were looked at, with motile abundance, sessile coverage, species richness, and various fish metrics investigated with GLM and MES post-hocs, whilst for multivariate, Ray Curtis was used for per-manova, simper analyses, and DB-RDA plots, of which R, primer, and event measure software was used for all of these statistical analyses. So, getting on to the interesting stuff, which is the results. Well, the first thing we can say is that on the boulders themselves, so at site and at boulder level, 110 different taxa were identified across the study period, and an additional 75 were found specifically in the rock pools themselves. So, for the site scale analysis results, specifically just for benthos, in total, no biodiversity indices were significant for anything, which shows us that there was no spillover effects present at our site, which means that the integration of these living boulders into the revetment does not enhance the biodiversity on the sandstone boulders surrounding them. However, for fish, we saw some pretty interesting results. So as you can see in these box plots, there was enhanced richness, habitat use, and borderline shoal size, specifically at the modified site. Although community composition was not significant, and feeding and behavioural data requires more data to be analysed properly, on the whole, this shows some pretty interesting results. So, at the boulder scale, which is looking at, first of all, the sandstone versus the living boulders, on the tops of the boulders, we can see an increase, a statistically significant increase in richness for the living boulders when compared to the sandstone boulders. Whilst on the sides, there is no difference. That means that we're able to conclude that water retention is the reason for enhanced biodiversity. And this was also the case for community composition. Additionally, when we broke this down further in the models, we can see that it is specifically the case for higher elevations and for sessile benthos. Regarding motile abundances, there was no effect, and coverage trends were very inconsistent with elevation. So putting this in the context a bit more of the key taxa and functional groups, first of all, our simper analyses didn't tell us a huge amount of information, only that there was enhanced oyster coverage on the sides of the sandstone boulders. However, we do have some interesting finds with our functional groups. So first of all, when looking at sessile benthos, both the sessile invertebrates and the algae responded and benefited from the presence of water-retaining features on the top of the boulders. Additionally, for algae, this was specifically red and brown phenotypes at mid and low tidal height. Regarding gastropod shell morphology, which is linked to resistance and dissipation, neither of the two, limpets or coiled gastropod shells, benefited from the study. So, now being able to look specifically at the effect of material. As you can see in this top left-hand corner graph, on the tops of the boulders, you see enhanced richness to the living boulders and the control boulders when compared to the sandstone. This is on the top, but there's no water-retaining features on the control boulders, so we're able to conclude that material is possibly a reason for enhanced richness. This was also the case for sessile community composition. Now, at the microhabitat scale, first of all, regarding rock pool textures, we found that there was no effect on any biodiversity indices. However, community convergence of these artificial rock pools with natural rock pools was observed, with no difference for motile community composition, and for settler community composition, only significant results of differentiation at 6 months. So in this DBRDA plot, you can see 6 months indicated by the round icons, and the blue is the modified size. And we can see at 6 months there's huge amounts of differentiation. However, by 12 months, when they're squared, we see overlap, which means that rock pool type, so whether it's an artificial rock pool or a natural rock pool, is no longer the main reason for the differentiation in community composition. So, moving on to my discussion, I'll start with 2 main points. Well, first of all, we can determine that there were distinct top surfaces on the living boulders due to rock pool microhabitats, specifically because of this water retention. Additionally, we can say that the artificial rock pools were indistinguishable from natural ones, which means the living boulders' aim in biomimicking natural habitats was successful. So in terms of the spatial scales, we can say that the implementation of the living boulders had positive localized boulder scale effects due to these rock pool microhabitats. However, for fish, we also saw preliminary results that are significant at the site scale, showing that increased algae coverage and growth may encourage herbivory behavior by fish. So, going quickly into the mechanisms behind all of this, well, water retention was deemed the most influential factor in our study. This adheres and aligns with previous studies, lots of which were done on seals, which also saw this effect. However, previous studies have also seen site-scale effects with benthos, but we didn't observe that. Regarding material, we saw weaker but present results, which also aligns with previous studies, which show that the effect of material is temporarily limited, especially once formal colonization has taken place. Previous studies have also shown that this is most significant for seaweed, and we also observed that in our study. However, there is one little issue with the study design that we have to take into consideration, that the pre-existing sandstone boulders will have had colonization already on them. So what we're seeing on the living boulders and the control boulders may not be a material preference, but just a preference for available space to colonize on. So, going on to rock pool texture at last, we saw no effect. However, previous studies have shown that texture is incredibly important in enhancing biodiversity. However, its role in rock pools is very much under research. So we need to better understand whether there is in fact no effect, or the issue is actually the design of the textures within the living boulder rock pools. Specifically that they don't meet the size-specific niches required by the local biota. Regarding the management, the integration of water-retaining devices to riprap can enhance biodiversity, but it very much depends on the number of units deployed as well as the position and the configuration of them. So in conclusion, integration of water-retaining devices such as the living boulders can locally enhance biodiversity, specifically on riprap revetments, and this adds to the growing field of eco-engineering and its ability to enhance local ecosystems. Thank you.
Thank you very much, and now we have time for questions. Who wants to start?
Well, I know I expressed it, so I can start just to break the ice. Congratulations, very nice presentation, nice work, nice slides. Just 2 questions from my curiosity, more than my knowledge on this topic. After that results, you say that first, you say that all the structures were in the project was started at April 2023.
Yes.
So when will you start to get the idea to make your master thesis with this? Because it's something that usually the students started in January.
No, of course. It's actually this whole project is run by a foundation called the Living Seawalls Foundation, which is partially funded by the Australian government as well as the University of Sydney, UNSW, Macquarie University, and a variety of different delegates. And they work specifically on creating modules. And this experiment was designed a few years ago, and I basically did the last, at 22 months, the final screening. So I did the collection of all the data and analysed all of the data that was previously collected by people working for the project who are no longer there. So, yeah.
Yeah, okay. Another thing, you say that for some species, this type of structure were not did not know any benefits, beneficial for those species.
Yes.
Are there other ideas to change the structure just to, because in some cases the impact is affecting some type of species in some areas,
Yes.
so maybe you have to change the design.
Yeah.
Have you any experience, or the group or the project is working also on that?
Yeah, the project, they're interacting with another university as well in Singapore. So basically these modules are designed by a company called Reef Design Lab, which works kind of internationally. And some issues that they're still trying to look at is which functional groups are responding positively to the design, as well as the issue of invasive species.
Yeah.
So at the moment they're trying to figure out whether the rock pool size, because they're just small rock pools at each one of these, whether that can be integrated and changed to see if it can be improved. We saw that the main biota in the area of this part of Sydney Harbour, the organisms that benefited most were the ones we expected, the ones that should be. They weren't invasive species that were doing it as such. However, it was really just a preliminary study. So they're going to, at the moment, they're in a project of redesigning it to go and see if they can add some different textural complexity and see if there's other ways to benefit specific species. I don't know if that answers your question completely, but there is a lot of work on it. So.
And in Europe, because I mean, this master thesis is in Australia, but you have been traveling about. There are other groups doing something similar in Europe. In
yes,
some areas,
there's one in the UK with Plymouth University and Southampton University. I think they collaborate, and actually some of the panels from Australia have been sent there and they've been installed there. So they're kind of working together. It's kind of quite a small group, but on the whole, I think it's a few universities in Australia, the UK, Singapore, and one in Hong Kong that work together on eco-engineering, specifically sort of concrete structures that can be integrated into coastal defences.
Yeah.
Thank you.
Thank you for your very good, very nice presentation.
Thank you.
I would like to ask you about the material and the texture. So you said in your discussion that the texture it was not influential to the colonization.
Yes.
But it is that. Do you expect that result, or it's something?
No, we don't expect that result. Previous studies have always shown that increased textural complexity enhances biodiversity because you've got crevices and protective spaces, and it also increases just the available space because of rugosity and available planar space, So it's really kind of odd. However, very few studies have investigated specifically in rock pools. So that's something that we need to look a little further into. So that's why we're questioning whether the issue is the design rather than, so for instance, a lot of the small motile organisms in the area might require greater textural complexity to be able to adhere to it. And also we saw that the rock pools accumulated sediment over time with the tidal fluctuation, which means that it might be hiding some of the textural complexity that we have in the rock pools, which ultimately means that it doesn't have an effect. So we need to differentiate whether it can enhance biodiversity, or in rock pools it's not really going to make a difference in the long term.
And what about the material? Which was, maybe I lost it.
It's, I believe it's called EC60. It's been previously tested a few times, and it's shown that it doesn't perform any worse than sandstone or any other sort of things. On the whole, some other sort of studies have shown that oysters tend to prefer it, as well as a few other sessile organisms. And this is basically what they're making all of these panels and stuff with across the world. It's kind of internationally recognised as a good concrete example.
And a last question, again. You talk about, in your results, you talk about the fish, and you said that it was very interesting.
Yes.
But how can you explain? So, why, in your opinion, there was this enrichment in fish?
Yes. Yeah, exactly. So maybe it's easier to show you an image of this one shows it quite well. So, compared to the sandstone boulders that now it's really hard to see, there's a lot more sort of algal growth and stuff within the rock pools and such. The coverage in the rock pools would be higher. So because of that, we're expecting on the side scale, we can see the feeding of fish also in the camera. We didn't have enough data to collect to analyse that statistically. However, actually I've been working with a little post with the thesis, and it's now, when we've collected some more data and I've analysed it, it's now coming also statistically significant, and it's for the amount of feeding as well as the grazing intensity. So what we're expecting is that the implementation of the living boulders in the riprap revetment increases the amount of available food and is encouraging from neighbouring areas fish to congregate and feed. So, yeah.
So species are fish.
Could you say that again? Yeah, herbivorous. Yes, specifically a type of fish called in the Prionus species, which is a type of surgeon fish, and those have extremely high grazing intensity. Yeah. Thank you. Thank you.
Yeah, maybe I'll end with a question that is more provocation, but what is the purpose of deploying that kind of eco-engineered substrate in areas that are highly artificialized and highly a bit more in terms of biodiversity?
Yeah, it's a good point. There's a lot of conflict in the area whether we should be spending our money on creating infrastructure like this rather than implementing like nature-based solutions and those sort of things. So, for instance, using mangroves or boulders as a way of enhancing our local reefs. But in a lot of places, this is not feasible, if that makes sense. So, for instance, in the harbour of Sydney, because of a lot of port activity, they're having an issue with this sort of erosion. So being able to create things like this is only a small piece in the addition. However, we hope that it can be something that is integrated everywhere. So when we're seeing more and more riprap creation, that instead of just starting with these retrofitting it, it can be made into the design of ripraps themselves. The same, for instance, with seawalls, that instead of designing something and retrofitting something onto the wall, the design and actual engineering construction can be altered. And that will help at least in terms of places where nature-based solutions aren't feasible, specifically in very highly urbanized locations. And of course, the point of this, you think, oh, why do we need to enhance the biodiversity in these sort of areas? Is there are a lot of course, benefits. So around Sydney, fishing is particularly prevalent, which of course that's not always the point of implementing these things. However, you can really, by enhancing the biodiversity, nature-based tourism is huge in the area. This actual implementation was funded by a kayak tourism group. They paid for the installation of these boulders because they go around and they come and show, and they email me constantly saying, any more results, things like this, because they're passionate about being able to show this to people. And in the whole, Sydney has a huge amount of eco-tourism, whale watching, this sort of stuff. So it's being able to do this can both help enhance things in terms of the environment and human well-being.
Do we still have time?
Yeah.
A few minutes. But you can ask further questions. You can ask us if you like.
So anybody in the public has questions?
I'm curious, I didn't know I can continue to work with them, or like some of the information that
Yes.
continue working a little bit.
Yes. Yeah, we did some sort of model alterations and added some other data. So I've continued with that. At the moment, it's put in to be reviewed for a journal of applied ecology. And yeah, I'm hoping to do a PhD in NUS in Singapore, which is affiliated with this project, and they have worked together in the past. So I'll continue on this, but it will be specifically also working on the design of, for instance, the textures and stuff, but also integrating both passive and active restoration. So transplanting animals and organisms such as corals and different macroalgae to be growing in it once it's placed in water. So it can both passively and actively enhance urban settings.
How much is cost wise? This is also feasible in more rural areas.
At the moment, the cost is rather expensive. For the creation of the boulders, it's going down in price. I believe at the moment, Sydney by Kayaks have the 15 deployed. The issue was, you saw previously, there's a crane and stuff that we have to use to get them in because they're so unbelievably heavy. So, you know, like I think it's really heavy, let's put it that way. So I think Sydney by Kayak paid something like $5,000 to have these 15 put in. However, at the moment, the New South Wales government has given, I think it's $3 million towards the implementation of these all across the city, and that's just for this, not just the panels. So with the production of these, which can be definitely upscaled, because at the moment it's just through one tiny lab that creates them, it's something that should be feasible, and worldwide we're seeing different governments contacting the organisation to do it. We also get a lot of sponsorship. So they were sponsored by the World Sailing Championships. Volvo sponsored them, and they created special panels with that, and they basically do a lot of fundraising to be able to implement these.
Any other question? I have one question, if I may.
Yeah.
So we can see good effect. What about bad effects?
Yes.
And then two maybe. Concrete is the worst material in terms of carbon dioxide release in the atmosphere.
Yes, yes.
What about this? And the second is that many of these places are occupied by microalgae.
Yes.
You did not tell us anything about it.
Yes.
And some of them are very toxic.
Yes.
Is that considered in the deployment, in large-scale deployment?
Yes. Yeah, it raises a good point, that whether we should be using artificial material to do this in the first place, and kind of the issue, of course, is the design of them. But I know that I've spoken to some people recently, and they're finding ways of designing it specifically with sandstone, being able to create them with sandstone. There's some sort of printing devices that they're creating now that you can actually do with sandstone. So they're looking into that, but at the moment the price of that is so much higher than that of the concrete ones. This design would be especially good for areas that, for instance, pre-existing riprap, which is already concrete. This happens in a lot of areas. So then you see absolutely no difference. But the fact that we're seeing that there was enhanced biodiversity even on the concrete ones, and it wasn't necessarily invasive, on the whole, it seems to be beneficial, even though it's an artificial material. And your second question regarding microalgae, yeah, that's something that's also been brought up, whether we're encouraging, for instance, eutrophication or algal blooms of toxic sort of organisms. I know that they've taken water samples at different periods to go and test that, to see if there's anything. I think they're kind of behind on all the sampling of it, but at least within the first year, there was no presence of any toxic microalgae. However, it's something that still needs to continue, since this is the first study that's ever kind of done like this. Yeah.
Thank you very much. One quick question.
So these are going to places where there already is a concrete structure, right? So
not necessarily. These are sandstone structures here.
is there any need to start putting in new, like when they build the infrastructure, they need to implement these sort of things
immediately
into the actual building, as opposed to putting supplemental things on top of that?
Yeah, this is something that at the moment they're trying to do. It's a lot of working with authorities and stuff to do that. I know that at the moment they're setting up another 2 projects for this, with one which is a new riprap revetment that the government was going to put in place anyhow. So it's contacting them before it's deployed to make sure that, we can do this before you even implement it. So to install these, they had to remove, I think they had to move some of the boulders and things like that. So it would be better to have it in the initial design rather than retrofitting. Yeah.
Okay, thank you very much. Also, the next speaker is Leo. We have 15 minutes, and I will stand up about 40 minutes.
Okay, everybody, my name is Cleo, and I'm going to be talking about my thesis, which is titled Participatory Activity and Conflict Mapping Inside the Maritime Space of the Atlantic Islands of National Park. So in 2022, the Montreal Biodiversity Framework was established, which sets to protect 30% of the ocean by 2030, including 10% fully protected areas. And this is aimed to be done through marine protected areas, which have the aim of conserving nature at sea. And they often have different levels of protection, including fully protected, which means that there should be no impact from extractive or destructive activities in these areas. However, in order to implement these MPAs, there are often a lot of trade-offs that have to be made between conservation goals as well as socio-economic impacts that these regulations can have. For example, on taking away fisheries, that can have an impact on local communities depending on resources in these areas. And a way to address this is through ecosystem-based management, which is essentially a more holistic approach of looking at ecological and socio-economic factors that are important in MPAs. And often this uses local ecological knowledge, which is knowledge that's not necessarily published in literature because it's really in a data-poor region, and it can be used because of people knowing things about these areas as they use them every day or are dependent on them. And a solution to address this trade-off and an ecosystem-based method is participatory mapping, which is essentially drawing maps together with people. And it's a very inclusive method because you're asking a wide range of people and stakeholders involved in the project, and you can also share these maps in a very easy and digestible way, and also for long-term engagement to continue such a process. And it could potentially lead to greater compliance, which is very important in an MPA in order to set them up for success. They're dependent on the people working there. So my case study is in the Atlantic islands of Galicia, which is along the coast of northern Spain, and there are a group of islands called Cias, Salvador, and Portugal. And in this study, I haven't addressed Portugal because it's, as you can see on the map in the right corner, it's very in the inner part of these rias, close to the river mouth, and has very different environmental factors and therefore stakeholders. So in this study, we look at the offshore areas. And this park was established in 2002, but it took 17 years to implement the management plan, and has to be reviewed in 2028, which is just in time for the 30 by 30 goals. And the park consists of 86% maritime space and has different levels of protection. And 0.4% of this maritime space is a marine reserve, but it's on the seafloor. So the seabed or the sea column, water column, is not protected. And on land, in contrast, it's 52% reserve zone. And a prior study that did ecosystem service mapping in the CS Island found that the greatest overlap between ecosystem services was found along the coastlines. So my aim of the study was to map human activity hotspots, stakeholder conflicts, and management perspectives of the national park to inform future management efforts. And I addressed these three questions. What are spatial patterns of high usage areas within the national park? To what extent do conflicts arise among stakeholder groups? And are these conflicts geographically concentrated? And based on local ecological knowledge, which are the areas that are most suitable for the expansion of marine reserves? So I did this by first defining my stakeholder groups based on the management plan and their authorizations that these groups face. So then I came to 5 groups: education and NGOs, recreational companies, which are mostly diving companies, nautical operators, small-scale fisheries, and researchers. And then I went on to identify individual stakeholders within these groups, mostly by looking at the national park website, because you need, for example, authorization to anchor or to dive. So there are companies listed on these websites that are able to operate here. And then for the researchers, I looked at literature that they published on this area and that they are working in local-based institutes. And I also used a method called snowball sampling, which is essentially asking people that are already in the study for other recommendations of people that they think should be involved in the study. And I invited them by email or individualized emails, explaining to them why they're important in this area or by just showing up to their piece of work. And I used an online survey tool called C-Sketch, which is a way that people can draw polygons inside of the park, and it allows to adapt questions to different stakeholder groups. So asking fishers, for example, more specifically about gear uses and target species, and divers about the depths that they dive. And these questions are actually linked to the spatial data that it's collecting. So the polygon's metadata includes the answers to the questions, so it's very easy to analyze. And then I asked about activities and conflicts, which are again linked to these individual polygons that I collected. And then also about management perspectives, and again, polygons for proposals of marine reserve, asking them to suggest a 10% protection area. And then in the end, I had 44 participants in my study, and 5 nautical companies, 6 NGOs, 4 recreational companies, 8 researchers, and 22 small-scale fishers. And I went all throughout the region of the park. And there's a little bit of a geographic trend in the south because that's where the biggest urban area is, as well as where all the research institutes are located. And yeah, and all the RIAs, or these states, are included in the study in terms of representation, which is important for the access of the people to the islands. And for data analysis, I made a lot of maps, everybody, with different levels of resolution based on their smallest polygon that was drawn, as well as a simple matter of certainty, because for example, for marine reserve, people were noticeably less certain about their answers, so I made the resolution a little bit less precise. And then I had to rasterize this polygon information, which should be 0 and 0. And the value is based on the ranked importance if people draw on multiple polygons. For conflict, is there a conflict? Yes, 1. Is there a conflict? No, 0. And then for marine reserve, the simple count of suggestions per raster cell. And then I standardized it between 0 and 1. And for the conflict potential, I separated it into levels of potential conflicts. And then for conflicts, I also looked at who is in the conflicts through directed network analysis to identify the actual people and not just the areas of conflict. And for the marine reserve, it was simple counts of marine reserves per cell. And I also looked at the ecological vulnerability and environmental priority. So I used a map that was from the Ministry of Environment of Spain, that is basically about the different habitats in the park based on the seabed mapping survey, and they included UNIS codes. And these UNIS codes I used to cross-reference them with the European Red List of Marine Habitat Vulnerability. So I created this map with classifications 1 to 6, which I assigned about levels of vulnerability from least concern to critically endangered, as well as data deficient areas. And then I use this to make an overlap between areas of high economic activity, which is fisheries, nautical operators, and recreation, and high vulnerability, which is near threatened to critically endangered. Because, for example, in this map, it already showed that in the north of Salva, there's a critically endangered, just one habitat of Cistera marina. And then go into the results. So these are the activities. And we go from the south to the north. So the CS is the most southern one and Salva the most north. And the lighter colors are the areas of higher levels of activities, and the darker ones of lower levels of activities. And this white box is the current marine reserve that's there. And you can see a clear trend of higher increased activities along the coasts and all through the islands, besides here, where there's also clear in groups of like small islands where you see levels of activities, and also more on the right side, which is the inshore side, so it's less exposed and more sheltered compared to the offshore side, which is more exposed to waves. So then you come back to the question, what are spatial patterns of high usage areas? And we find these trends, which are also found in literature on the previous study, but also around the world in terms of MPAs, that coastal areas tend to have higher levels of activities, especially also nearby easy access areas, so in the south of CS, as well as this trend of close to small islands and rocky reefs, which could be potentially related to the fact that these create an increase in habitat complexity, which is important for a lot of stakeholders in the area because they're dependent on fisheries or study biodiversity in these areas. And also because in Galicia, a lot of the fisheries is actually shell fisheries, so they need these substrates to get their target species. And specifically, the goose barnacle was most named as the targeted species in my study, and it's very economically valuable and important to everybody in this region. Then we go to the map of the conflict. So here again, the lighter colors are higher levels of conflict potential, and the darker colors are less conflict. And actually in CS is the only part where there was a critical conflict potential reached, and it was specifically in Rodas Beach and Venus Island, which are areas of high tourist activity, and they're quite famous areas as well around the world. And again, it's more on the inshore side, so where people are easily accessing and more sheltered. But then the question is, who are in this conflict? So this is the network analysis, and on the top we have the abbreviations, and there were actually 3 more groups identified by people or named by people's negative impacts in their conflict. So it would be bateros, which is the mussel seed collectors that collect mussel seeds for aquaculture inside of the RIAs. The ferries, so people taking ferry operators, and bureaucracy, so any legislative input. And you can see that the conflicts are, the darker red ones, are concentrated around nautical operating and small-scale fisheries, and they even have loops inside themselves, which means that participants that are part of these groups name their own group as a negative impact on their activities. So you have within group conflicts even. So to come back to the question, to what extent do conflicts arise among stakeholder groups, and are these geographically concentrated? There are definitely conflicts, and they're more so in the south and on the inside, so nearby easily accessed areas as well as tourist sites. And this could be potentially linked to the fact that fishing is happening inside the entire park, so that's why these conflicts are mostly related around fisheries, because they are occurring everywhere, as well as the notion of simple conservation versus kind of fishing clashes which happen in the park, which is often the conservation stakeholders named fisheries as a negative impact. And then again, close to the coast, there's a lot of fishing activity due to the shell fisheries, which is this high area of activity. And this is a quote from one of the interviews about anchoring. So there's no real control, just anchoring permission. However, there's no real surveillance regarding the anchoring. So there's this real notion of that this anchoring of both nautical and fisheries can have a negative impact on the seabed, even if they use these moorings, which can also have changed, disturbing the seabed. And then if we go to environmental economic priority, here the underlay lab is the economic map, so the fisheries, the nautical operators and recreation. And in orange are areas of overlap between high levels of economic activity, so 0.75 and up, and high levels of vulnerability, so near threatened to critically endangered. And you can see that there are some areas of overlap. And going here in the north, it's a very tiny area, but this is the area of the critically endangered seagrass meadow. So here there's already overlap between economic activity and this extremely vulnerable area, and that's actually where the ferry enters the island. And then in the south here, this beach comes back, Rhodes Beach, which is an area of sandy substrate, and this would also came as an area of high overlap. However, this is also an area that, because the UNIS classification was not always as specific in every part of the map, there's also a lot of data deficient habitats that overlap here, so it's important to go further into that. And then here are the marine reserve proposals, and an important note here is that a lot of people didn't want to draw a marine reserve actually, and for different reasons. For fishers it was often because they didn't think it was necessary or because it was negatively impacting their activity according to them. And for researchers or some other operators, they often said they did not have enough information or data to say such a statement about what to put in a marine reserve. So they have different reasons for not placing it, and they sometimes placed it outside of the park instead. But the people that did decide to place it, they mostly placed it here in the northeast of Salvador. So, and this is, if you remember the other, not a very active zone in terms of activities, and people often gave the reasons of placing their marine reserve proposal because it was outside of high activity zones. But also often people said that this is an area of vulnerable species because in Salvador also there was a critically endangered area there. And then also again Venus Island, which is an aggregation zone for several species such as skates and also close by beaches that have juvenile species occurring. So this was a reason of vulnerable habitat and species for the placement of this marine reserve. And it's close to the current reserve, which often people wanted to expand instead of having a new area. So then coming back to the question, based on local ecological knowledge, which areas are most suitable for the expansion? So it's areas in the north and close to current reserve. And this was also interesting that people provided alternatives or additional measures to marine reserves because they didn't think it was effective enough. So for example, higher prices of fish coming from the park, and also reduction of fleet size through generational retirement. So instead of giving your boat to your kids, having people buy the boats. And then we also found an overlap or kind of a match between information in literature and the maps, as well as polygons drawn by people specifically for these three areas, which are about one study is done based on the mapping of each of these habitat. And this is a picture of Merle, and they found that we found a match between where they were mapped by people in the study and also in the literature. But the vulnerability needs to be addressed a little bit further in terms of how vulnerable these habitats are by this activity. So in the end, this study was the first participatory mapping study in CS, ONS and Salvador, and they found a trend of these coastal hotspots that's in other areas as well. This is a unique combination of both activity, conflict and marine reserve expansion, which is not often in participatory mapping studies. Usually they focus on one thing. And we found that it's somewhat of a match, but it needs to be addressed further between ecological info and local ecological knowledge. And then it's also going more deeper into the conflicts. And the next step would be to share these results with our stakeholders and show what's going on, and maybe continue some next steps in participatory mapping, expand the assessment of ecological information, as well as continue with a larger set of stakeholders as more groups were named in the conflicts that were not included in the study. And then in the end, we studied spatial social dynamics to set an MPA up for success. And this one, my acknowledgements. Thank you for listening.
Okay, time for questions.
Yes.
Thank you, Leo, for your presentation. It was very good. I really appreciate the way you present the data. And just a quick curiosity question. So, do you propose the questionnaire?
Yeah.
Because it was you proposed to stakeholders and to like companies. So the one you talked at the beginning. Do you find any like people that want to participate? So they are all involved in this stuff. So they are all happy to do this, or do you find some
Reluctance?
complaints about people participating in this kind of mapping?
Yeah, so definitely not everybody wanted to participate, also because we emailed them, so you can really be easily ignored. But some people, especially fishers, did not feel the urge to participate, and we didn't contact fishers directly, but we contacted technical assistants in local fishers guilds that are like the bridge between science and fisheries, and they often also responded like, it's nice, but nobody wants to join these studies. So that definitely happened, and then some people explicitly said no, but usually the companies, like sailing companies and diving companies, were very interested in participating, because in the park there's a lot of ecotourism, so they tend to care a little bit about what's happening, because they need these authorizations to even go there. And then we also tried to contact the mussel seed collectors and the ferries, but they never replied. So they were contacted, but they were not willing to be involved.
Because the question was, I think, very difficult, so I'm wondering if everybody can also understand this.
In the survey, you mean? Yeah, no, we made it sure that the survey did not use like super biological terms, so words as conservation and stuff like that were not used. It was always about ecosystem health, or like threats, or things that are affecting them, and personal connections. It was meant to be digestible, and then for the researchers, the questions were slightly more in depth, because they are also a source of information that maybe they're working on something that's not yet published, but they know that they went diving somewhere where they've seen seagrass, but it's not their scope of studying, but they know there, and they sometimes also use that to draw a polygon.
Okay, Cleo, beautiful slides. I mean, I love Galisa. I have been there as a visitor, so I'm part of one of the guys that was there as a tourist in
Yes, probably, yeah.
Did you have the opportunity to visit?
Yeah, so all the pictures are made by us, the people in the lab, and by me, that you've seen on the slide, so we went.
Okay, I was, first question, maybe it's more philosophical than anything. How we control that what the people is saying is true?
Yeah, so that's something in terms of their activity, you mean? Like where they go, or the general ecological information? I would say that this is simply, how do you say that? For ecological information, you can reference it with potential literature, but that's kind of the problem, that the whole park is not as covered in terms of, how do you say that, publications. And for other people, if you really want to go into it, it would be by GPS tracking on boats and everything, but then people are less likely, I think, to participate in these types of studies because they're feeling more controlled. So it's also by kind of seeing if people's perceptions of other stakeholders match. So for example, sometimes during interviews, divers would say there are a lot of boats here, or nautical operators, and then nautical operators also say there's a lot of boats here. So it's kind of like combining this information, but this is a pilot study I don't think that this should be the reason to draw a marine reserve here because people don't go there. It's like the first step to go in depth, but I would say GPS trackers is like
Yeah,
the
because
main reason
you
to
remember
track it.
yesterday from Francesco that what the people say is not exactly what they are
Yeah.
doing. They are not fishing in the place that they are saying they are fishing.
Yes.
And it's crazy.
That's definitely true, and also the people said a lot of poaching and illegal fishing is happening, which obviously they're not going to draw. But then because we have quite a wide range of stakeholders, there are other groups that provide this information on each other, kind of. It's like more of a, because so you need everybody involved to have kind of this closed circle of information.
Did you attempt any more active method to search participants, or how do you think that, for example, if mapping, doing what you were doing for this last 6 months is important, how do you think that we can improve the participation to have real data with all the stakeholders that you really need to get conclusions that really can help to establish the marine protected areas and establish the...
Yeah, so the main thing that we did is we went to them. Like, we didn't ask these people to come to us, like
Because
we went to their place of work. Yeah, that's the invitation, but so it's also by calling, and if people didn't reply, and I was in the area for another interview, I showed up. We went and we just rang the bell, and sometimes they were like, well, this is a bit scary, but often they would just be like, oh, come back in 2 hours, or I'm doing it right now. So it's by showing up basically to their place and making it as easy as possible for them to attend. And even for researchers, which are really busy people, I'm sure you know, we made sure that they could do it online. So because we understood that they are a little bit more easy to understand these type of studies. So for them, it was also the option to do it online, and we hosted a workshop where they could come at any time. So it's by accommodating, I think, the needs and making it super accessible to join.
Just the last question. Sorry, I love this. Did you make different maps for different stakeholders to compare? Is it kind of statistic or something that you can do to just...
Yeah, so I did. I also looked at the different stakeholder groups independently, and then there's something called, it's really general, geostatistics. It's simply looking at the overlap. I looked at overlap between each group and also where the different places go. And I corrected these polygons also for the area, because fishers were always like, I go here. And the divers were like, actually dive in this 1 centimeter square area. So you can see really big differences in the size. I also have independent maps of where everybody goes, and it's really also dependent on the environment, like the physical environment, because it's very different, like the exposed and the sheltered side, like it's completely different. So, and people, you cannot dive always there. So that also explains a lot of their placement of polygons.
Thank you so much.
We still have 6 minutes.
Yes.
Before clapping. Okay, thank you very much, Kim, for your nice presentation. Just a few questions. If I understood well, there's already a reserve.
Yes.
Is that. What about his local perception and its ecological efficiency so far?
Oh yeah, so I actually looked at that, but I didn't include it in the study. I also asked people about their perspectives on the current management efforts that are being made and the threats that they face. And the marine reserve is on the bottom, so you can fish on top, which is, I feel like, step 1 of the critique that was made. So that was mostly addressed, and also the sheer size. It's very small, and it's also in an area where the substrate, like, it doesn't necessarily cover a lot of habitats, which are known to be ecologically vulnerable or harbor. Like, there's a lot of biodiversity in this area, and a lot of like nursery sites and everything, and it's not necessarily there where the marine reserve is happening. So the perspectives of the people working in conservation were very much like, we need to do a little bit more than this right now. And then for the fishers, it was super individual. So some people said, take it away, but it's already there. And some people said, don't put it in my place of work. And others were like really thoughtful and were like, actually, I know that there's a seagrass meadow here, I've seen it. I would like to base it on there, and there should be a bit more spread out between the islands so that it's equally affecting all the fishers in every different region. Yeah, so it was super individual for the fishers.
Okay. Can you tell us more about the way the vulnerability was assessed in your...
Yeah. So here I used just the Uniscode. So this was just a simple first way to look at vulnerability because I didn't think it could be ignored. And I used a study or like a publication from the European Commission on habitat vulnerability. And there's like, they have a classification of not vulnerable, so that's their classification. And then they update the vulnerability like every couple of years. So I used the most recent one, which is from 2022. And then I cross-referenced it with the Uniscodes that were in the habitat map, but they were not always as specific because if you're aware of Uniscodes, they can be with multiple numbers after a decimal. So, and that defines like how specific they describe a habitat. So for some parts of the park, it was only 1 decimal point, and for others it went to 3 or 4. So I had to generalize it for them, and that's why I'm talking about the fact that it's like not completely representative of the vulnerability, because it can summarize a habitat that's maybe not there because the classification is too general of that specific area, but the difference between the park or like the habitats in the park, it was not consistent. So yeah, I would say that for the next study, it should be based more in depth on like the literature that's there. I started doing that a little bit to match it. As well as simple, there's not known active monitoring and happening. So I would say that's also would be a way to address it more in depth. But this is just the first initial way of looking at it. As well as cross-referencing it with more indexes, such as like in another study they used IUCN red lists, but because there's not that many publications and I didn't want to bias it for certain species that are studied a lot, for example, because they're fish a lot, like the octopus, there's a lot of information, but then you're missing a lot of species that I don't know that are there. So usually they also use species richness indexes and stuff to assess the vulnerability, but I didn't have the time and also the information to do that here. So this is just the initial way of thinking on it.
Did you consider the fact that some habitats may be vulnerable to some kind of anthropogenic pressures, but not to others?
Yes, for sure. So for example, diving is very, it's definitely an anthropogenic activity, but the impact of diving is way less than dropping an anchor in a seagrass meadow, and also the behavior of divers is very different because it's often people that are quite aware or willing to learn about marine environments. So for example, this island is highly fished and highly dived and highly researched, but the impacts are completely different of like the people that go there. I would say the fishing pressure is very different than a researcher studying an area or a diver attending it. So yeah, that's very different. So that's why you need to know who's there, not just all the people that are there.
And just the last question, on the map that you showed about human activities.
The first one?
Yeah. Yeah, it seems to me that you have much more data for the
CS Islands?
Yeah.
Yes.
And the other one, there's just a few polygons, so you
Yeah.
think that it's just one person who draw this polygon, or at least a limited number. How exactly is it imbalanced?
So that's why in my methods I explained that most of the people are in the south. So it's like this trend can be explained simply by the type of access that people have to the islands, because to go from the southern island to the north it takes like 3 or 4 hours. So if you're a fisher in the south, you often do not, some do go to the north because it's very small boats. So it's also reflective of the amount of people that are there. And CS Island is also the island that faces the most tourists. If you go there, it's just people that are there. So I would say that there are definitely more people that we interviewed that are based there, but in part it can simply be explained because it is visited the most, and the northern part is less populated and less known by tourists. So yeah, but every island, for example, the nautical operators, we had 2 here and 1 there and 1 there, and the divers, like in every island we had at least 1. So it's definitely at least 1 per group. Yeah, so that's fine.
Thank you very much. We'll take one question.
It's more of a curiosity for the questionnaires. Did you find like any problems with the different stakeholders like responding to the whole questionnaire? Where did they have to take it for you to take it into consideration?
So they responded almost all to the whole questionnaire besides the marine reserve part, but then I gave them the option to draw something. So I have this map actually. To draw something outside of the park. So some people were like, I don't want them to be in my park. So I asked them, okay, if you want to place it anywhere else, where would you put it? And sometimes it was researchers being thoughtful about like connecting things and different species that are there. So I provided them with an alternative. But for all the other parts, people answered everything. So I did not have problems with having to take out things because they weren't answered, besides the marine reserve. And that's also why I don't claim to be like, this is the best area for a marine reserve, but this is what people that answer these questions are saying to be to them a potential area for a marine reserve.
Thank you.
Thank you very much. Now there is half an hour rest, and then you can go with any of your colleagues.
questo? Sì, sì.
Thank you. Ready? Thank you. Okay. It's time, so welcome everyone to the second session of Group 4. The first thing is congrats to all the 3 excellent master thesis presentations that we will have now. I'm Gaisel Borrell from the University of Oviedo, and I will be initiating this session. I will have a great company with Alexander Novici from the University of La Marche, Lars Sterman from the University of La Sorbona, and Karim Ersini from the University of Faro, Portugal. Remember that you have 15 minutes for your presentation and then other 15 minutes for questions. 1 minute before the end, I will start. Okay, just to let you know. Enjoy.
So, hello everyone, I'm Paul Dreger, and today I would like to present you my master thesis on improving EDNA detection of the sponge natural sampler Aplyzina elephoba in an integrative approach. So, sponges might be quite unassuming organisms at first, but they're indeed key components of benthic habitats. They provide complex 3-dimensional structures that foster rich biodiversity, not only around the sponge, but also within the sponge's canals and its tissue. However, this structural complexity, of course, makes it difficult to assess this biodiversity using traditional laboratory or in situ methods. But sponges are not only important providers of food and shelter, they are also capable of filtering enormous amounts of water, meaning they accumulate all kinds of particles from the environment. And this includes environmental DNA, so genetic traces from the water column from all kinds of organisms, which we can utilize to characterize the surrounding communities using genetic barcodes. The idea of natural sampler DNA from sponges has already been tested in some studies over the recent years. However, most of these have focused on fish. The reason for this is that if we use universal metasome metabarcoding primers, we'll of course primarily amplify the sponge rather than the eDNA, which is what we're actually interested in. A potential solution to this could come from gut content analysis, where we also want to suppress the predator DNA to enhance prey DNA detection, and this is so-called blocking primers. The approach is very simple. We're just adding an additional primer to the PCR mix, which instead of amplifying, blocks the respective target sequence. However, this approach has never been applied to the concept of natural samplers to date, and this is where my study comes in. I wanted for the first time to evaluate the effectiveness of blocking primers in the context of natural samplers, and for this we selected the sponge Aplyzina elephoba. Aplyzina elephoba is a very common sponge throughout the eastern Atlantic and the Mediterranean that also hosts rich fauna in its canals, making it also a key species for restoration efforts. And so we wanted to use the metabarcoding data from using the blocking primers then to define the fauna communities associated to Aplyzina elephoba, using them both the molecular methodology as well as morphology analysis to achieve the most comprehensive biodiversity assessment possible. And thirdly, assess the spatial variability of those communities we identified. So we started by collecting whole Aplyzina elephoba specimens at 7 sites around the Canary Islands in April 2023 and November 2024. At each site, 2 to 4 samples were collected opportunistically by covering the sponge with a Falcon tube and then cutting off the sponge at the base, meaning we're not only sampling the sponge, but also its surrounding water. All samples were collected at a depth of 14 to 30 meters, so all located on the shallow island platform. Each sample was then processed both for morphological and molecular methodology, meaning one piece of tissue was set aside from each sponge for an SDNA metabarcoding, and the rest of the sponge was then cut up to extract the associated fauna, which was sieved by 45 micrometers and then classified to major taxa. For the meta barcoding, I first needed to start by designing my blocking primers. We selected the CO1 gene as our target region, which is a very common universal metasomal marker. And for this, I designed my two blocking primers specific to the host Aplyzina elephoba. One was an annealing inhibiting blocker, meaning it was supposed to prevent the binding of the universal primer to the Aplyzina elephoba sequence. And secondly, an elongation arrest blocker, as the name implies, handbraking the process of elongation through the base addition to the sequence during PCR. The design primers were then tested in silico with software called E2PCR to ensure that they only bound to Aplyzina elephoba and did not have any impact on other organisms. And then modified with the C3 spacer, which is then the actual modification that turns the primer into a blocking primer. For amplification and sequencing, I then prepared 3 treatments, one for each blocking primer, so one for the annealing inhibiting primer and one for the elongation arrest primer, which were each added at a ratio of 1 to 10 to the universal primer, and then a third control treatment to then evaluate the blocking primer performance. All 3 treatments were then amplified by PCR and sequenced with nanoporous mini-ion. I don't want to go down too much into detail into the sequence data processing, but I wanted to highlight one particularity of nanopore, which is the building of consensus sequences. Nanopore has a slightly higher error rate than Elounda. So we built consensus sequences to reduce the error rate that was introduced during sequencing. Here we used a clustering percentage of 90%. And the consensus sequences were then that was used to build the OTU table and assign taxa. The taxonomic table was then transferred into R and decontaminated, meaning all organisms from the controls were removed, as well as anything which we're not interested in, so in this case anything but metazoa, to give us the final OTU table. For morphological analysis, we simply classified and counted all of the associated benthic organisms with major taxa and then standardized that to 10 milliliter sponge volume. For the data analysis, I wanted to compare the 3 treatments, and my key question was, how do the blocking primers impact the Aplyzina read count, and how does that in turn affect the OTU count after decontamination? Those were my 2 response variables. The most successful blocking primer based on the prior analysis was then selected for analyzing site-level differences alongside the morphological data set. First by community composition for each taxonomic rank, so to improve the comparability of the molecular and the morphology data set, we separated the taxonomic ranks, the phylum, class, order, and so forth, as well as by diversity indices. However, that will not be discussed here today, but it is included in the work. Coming to the results, we actually observed significant effects of the annealing inhibiting primer, which led to a reduction of Aplyzina reads by about 55%, which in turn increased OTU count per sample by 97%, which you can also see on the right side in the box plot with the retained OTUs after decontamination on the y-axis, with the annealing inhibiting primer on the left side, performing significantly better than the elongation arrest primer as well as the controls treatment. So we used then that data set to perform the community analysis based on natural sample DNA metabarcoding. What we can see here is that based on mean relative abundances, for examples merged together, based on read counts, chordates are the dominant taxon across almost all sites. Only one site stands out here. GAR in the middle shows very high arthropod abundances. This was largely based on one single sample that was almost exclusively dominated by Tecostracum, so barnacles. And this points towards something interesting which you can also observe in the other samples when we look on lower taxonomic levels, is that most of the organisms we identified to lower taxonomic levels were actually epibiotic. So Acidia, for example, or Parazoantes, as well as Tilodina perversa, which is a very common predator of Aplyzina elephoba. However, many of the measures we could also not assign at all to lower taxonomic levels, unfortunately, including almost all of the coordinate sequences. And if we now look into the principal coordinate analysis, here based on OGU level, on the left side Faulkner-Curtis dissimilarities and the right side Jacquard dissimilarities, you can also not really observe any meaningful pattern here. The variation of the principal coordinate that is explained is very little. And if we now look at the morphology, we get a very different picture. Now, the community composition is almost exclusively dominated by arthropods, being about 80–95% in almost all of the sampling sites. These are mostly amphipoda, isopoda, but also harpacticoid copypods. Only one side stands out, which is out here on the right side, which you can also observe if we now look into the community analysis based on the principal coordinates based on morphology. We can see that again outclasses separately from the rest due to its high abundances of annelids and mollusks, whereas the other sides are largely defined by their arthropoda, particularly amphipoda and isopoda. Here, this unclassified might be a bit confusing, but it's just all organisms that were not classified to the respective taxonomic level. So what can we take from this? We saw that the OQU count was almost doubled upon the addition of the annealing inhibiting blocking primer, whereas the elongation rest primer did not really show any notable effect. This aligns with findings from gut content analysis, where we also know that the annealing inhibiting primer usually performs better than the elongation arrest primer. And if we look into the community, we saw very different pictures based on both methodologies, with both methodologies complementing each other, with an SDNA metabarcoding, with identifying epibiotic macrofauna, such as Tecostraca, Pausomtidae, or Acidiacea, whereas when we looked into morphology, we found mostly endobiotic, myo, and macrofauna. These were particularly amphipoda, isopoda, and harpacticoida. We also saw that one side was clearly distinct, was characterized by a higher relative abundance of annelids and mollusks. We did not take any environmental parameters in the study, but a potential explanation could be that crustacea are more sensitive to hypoxia, which could, for example, be induced by sewage pollution, whereas annelids and mollusca are not as sensitive to that kind of pollution. However, this will require attention in further studies, as we did not take the data required for that analysis. We also observed some limitations in the study, with bioinformatic analysis being hampered by very low numbers of reads that were assigned, particularly the coordinate sequences. We could not really assign to taxa mostly. One explanation for this could be that I selected relatively low clustering percentage of 90%, which potentially could have led to an over-merging of the sequencing, so that we had a reduced taxa richness eventually. I also used rank-specific identity thresholds. So for each taxonomic level, I identified one percentage identity. If that threshold was not met, then I went up to the next higher taxonomic level to ensure that the taxonomic assignment was actually confident. But of course, that is very problematic if you work with such a diverse group, such as Metazoa. So probably we'll have to look further into that for future studies. What we also found was that many of the organisms that we identified were actually epibiotic, potentially being directly associated with the sponge, so meaning growing on the sponge. And this raises the question, what was actually the environmental DNA from the water column? Of course, we had a lot of coordinate sequences, which could potentially be unidentified organisms that were coming from the water column, but the explanation for a reduction in EDNA detection probability could be that we use a longer fragment for amplification. We used 600 base pair fragment instead of what is commonly used for Illumina barcoding, which is usually like 150, 300 base pairs. And as the proportion of amplified fragments decreases in the environment with amplicon size. This, of course, might mean that we had a reduced number of environmental DNA. Finally, key learnings. What can we take away from this? Blocking parameters are indeed an effective tool for mitigating host DNA overabundance in an sDNA metabarcoding. We also gained first insights into the associated fauna of Aptesina elphoba on the Canary Islands, laying the groundwork for further studies. But we also observed, of course, need for further research, particularly the bioinformatic processing will require some further optimization, some adjustment of parameters to optimize the taxa yield, as well as looking into the effect of blocking parameters on non-host taxa. We assessed that in silico. We didn't actually assess that in vitro. So this will be important to ensure that there are no biases introduced by blocking parameters, as well as the identification of factors that potentially could hamper eDNA detection from natural samplers. I mentioned the marker region, but of course it could also be interesting to look for other natural sample species, spawn species in particular, but of course also other filter feeders could be possible. Finally, I want to take the time to thank everyone who made this project possible. Foremost, my supervisors, Federica, Marina, Martina, and Alessandro, without whose mentorship this thesis would have not have happened, as well as everyone who participated in the sampling activities and helped me with the laboratory work, as well as Professor Sergio Rossi as principal investigator of the Ocean Citizen Project, of which this research was part. Thank you for your attention.
Thank you very much. Thank you. You were on time, so no need for advice. 1 minute. So, two more.
Okay, thanks. Very nice presentation. So I have one question about the method first, and then about some results. For the methods, I'm not a specialist of EDNA and sequencing. So in your slides, I could read that anti-blocking primers, they were predator and prey.
Yeah, that's
Can you
because...
explain a little the functionality, why did you call that predator and prey? And,
in this case, I used a graphic from a previous paper, which used blocking primers, but these studies on blocking primers have only done for content analysis. So that's why here I left the graph as it was.
why do you call that predator? Who is a predator? And who is a prey? Why do we call them?
Sorry. So, I think, for example, this study was in particular, I don't know if it was fish or shrimp, but let's say, so in this case it would be fish. There was the predator eating the shrimp, and we want to analyze what is in the gut, what is in the stomach. Often it's too digested to identify anything. So we want to remove the predator to improve actually the detection of what prey is found in the gut.
Then I was surprised between the difference between the morphological analysis and the genomic analysis. In the genomic analysis, Corata appears first. The other one, I forgot. You didn't see you could not, why in the morphology you didn't see the
That is a good question. We also saw in the morphology, so morphology also had, so for example, we had Tilodea perversa also in the morphology. We didn't have Arcidia or Tecostrata in particularly high amounts.
how is it possible? It's the same sample.
The reason it's possible is because if we have if you, for example, have a single layer of Elounda inversa, is enough if there's no EDNA to dominate the recount. So if there's barnacles sitting, even if it's just a couple and they are this big, they will dominate. And the reason is that we didn't really see, because we had almost no amphipoda in our NSDNA metabarcoding results, which is of course a bit surprising because they are like in almost all of the samples they're the dominant taxon. But of course, you're also targeting two different communities, one inside the sponge and one outside the sponge. So,
and could the reason be that here there are counts, that's proportions on counts, it's not on biomass? Is the EDNA more related to biomass than the count?
Yeah, that is correct. No, that is. That was what I was trying to say. If we have like 1 kilodynamic inversa like this, that is of course a problem with metabarcoding overall. We can't really, still like very difficult to quantify, and although there is some correlation with biomass, it's different between organisms. We have different shedding rates. There are people trying to do it for fish, but it's still we kind of need to transform, and we need to look particularly into correlations for fish in this case. So yeah, that is the problem with EDNA. So I feel like this graph was meant to give you an idea of how the recounts were distributed, but of course it doesn't give any quantitative indication of the community. Thank you, Lord. Alexander?
Yeah, could you comment the fact that the morphology data show a pattern while the DNA barcoding is not showing any pattern in your graph? This one.
Yes.
I have no idea.
No. I'm just thinking how to answer the question best, because this is based on operational taxonomic units, which are, of course, they are always a bit overestimating, or that's the kind of the consensus that they tend to overestimate the actual diversity that we have in a sample. As they basically just kind of clustered on similarity. They are not necessarily one taxon. And the reason could be that we here had not only an over-merging of consensus sequences, but after the over-merging, so I didn't go too much into detail on the sequence data processing, but what we did actually was we first clustered all the reads by 90% similarity, and then reclustered them to OGUs by 99% similarity. And if the clusters were too big, they were only subsampled to 500 sequences. So this could potentially result in that we have
what do you want to say?
No, I wanted to say that if we have a lot of OGUs which are very similar in their taxonomic identity, so we don't really observe any meaningful pattern. Yes, that is. I think using OGUs is a bit problematic, as I didn't really identify much to lower taxonomic levels. That was the one I had. So, but yes, it's true that we ideally we would have observed at least some kind of pattern. But also, if we actually looked into the tables, we see that they are also within the sites. They're very inconsistent, like what reads we find. So at least for presence, absolutely would expect some sort of pattern. But of course, if we don't really have that many taxa, so for example, for phylum level, I think we had 10. That is not enough to achieve any clustering if we have presence absence. Yeah.
Okay.
So with the morphological identification, those are the things that you actually find on the sponges. But with the EDNA, you are sampling the water, so you could be detecting very rare species that may have nothing to do with the actual sponges. So how do you think these two methods are compatible with your, how do you see if you want to study the communities of sponges, do you think EDNA is the way to go, or is there still room for a lot of improvement?
I don't think it's possible to use EDNA metabarcoding as standalone at this point. I think we always, for this data in particular, I think if you want to look into invertebrates, we will always have to use a combined approach of morphology and metabarcoding. I think, for example, from the environment, I think if you looked at the fishes, in the fish, then I think metabarcoding could be a very good approach. There we also have very good convergence of identified species from morphology and from metabarcoding, with metabarcoding even usually identifying more species. So thanks to many studies that used this natural sample approach worked in the deep sea. We often have, it's not very easy to sample and also difficult to detect rare species. So they can be very helpful to just use the EDNA metabarcoding as a means to identify which species is from the tissue sample. But I think in this case, I think we will always rely on a complementary approach with
the EDNA from the sponges, you would be sampling the water just from the very bottom, not the...
Yeah. Mm-hmm. But if what I didn't mention is that of course, with EDNA sampling, we have very much constraints through filtration and preservation, which of course, if this works well, and this depends of course on the sponge species, we can circumvent and improve the detection ideally. That was one of the original ideas. Many of the EDNA sampling methods are usually more sophisticated, very high-tech solutions or using plastic EDNA samples, but if this works, it could be effective.
More questions? Any questions from the floor? Or yes? You? No? Okay.
Yeah, I have one question, but there is a question here.
No?
No, so
Okay, one.
What is a different species that you think would be using if this species doesn't work? Why different species might not be able to work? What different species they use?
Like different sponge species, you mean?
Yeah, or something else you can use.
Well, there have been studies on different sponges before, so there have been targets on different genera of sponges. And one thing that was observed is that in tendency, low microbial abundant sponges are more suitable because they have a higher pumping rate and at the same time, of course, decreased metabolic activity in the tissue because they have less microbes, so less risk of EDNA degradation. And Artesia foca is actually is a high microbial abundant sponge, so it might not be ideal species in this case to use as a natural sample as itself, but of course as it also hosts a very rich fauna, and in this case the Ocean Citizen Project is like one of the key species for restoration efforts. This was still interesting to test. Yeah.
It's not a bad idea. I mean, I work in environmental DNA and we have 1 minute, but so two fast questions. One, did you check that the species that appear in your morphological analysis have data in the databases? Because in metabarcoding, the problem is that you compare everything with the database, and so if the species is not there, you won't be able to.
It's actually, like, I didn't mention it here because it's kind of like a generic thing usually in metabarcoding, but actually as we also work on archipelago, we have a lot of endemic species as well. So we have a lot of species, like I think it was from 2020, but there was one paper which said we have about 34% of macroinvertebrates covered. So that's
maybe an approach to try to complement the databases with the local fauna could be helpful, and also to change the marker. I mean, again, the problem is that you are using nanocores, so long fragments. You can use the complete marker, I mean 600 base pairs, but maybe you need to reduce the fragment and change the methodology for sequencing.
Yeah.
And with that, maybe sponge are good samples.
Yeah.
Who knows?
Depends.
No? Thank you so much, Paul. Okay, the next one is Alessia. Remember the answer? Please, the pen. Are you ready?
Yes.
The floor is yours.
Okay. So good morning, everyone. I'm Alex, and today I'm going to share my thesis experience focusing on coral bleaching in French Polynesia. So, as we all know, coral reefs are incredibly important ecosystems. They are biodiversity hotspots. They're biogenic structures that are built by corals within 30 degrees from the equator. And they thrive in very warm environments and support billions of people providing ecosystem services, and they rely on a symbiosis between the coral and the zooxanthellae. So zooxanthellae are microalgae that provide up to 90% of the energy needed by the coral. However, this symbiosis is threatened by a phenomenon called coral bleaching. So this happens when the coral is stressed and expels the zooxanthellae, and the tissue of the coral becomes paler and up to becoming completely white. This is caused by several stressors, so anthropogenic stressors and also climate change. The main stressor is temperature rise. There is also agricultural runoff and herbicides, and then ocean acidification, overfishing, and plastic pollution. And this thesis in particular will focus on temperature rise. So this thesis was part of a larger project called Toa Aroa, which is inspired by a paper that was published in 2020 in Hawaii. This is a color reference card that aims at engaging citizen scientists and the community, and this allows people that don't have a scientific background to detect early stages of bleaching. So we tried to do the same thing in French Polynesia, aiming at empowering the local community, and this was done in collaboration with a local NGO called Reba Atea. So my thesis was setting the first few steps to create this color gradient, and I tried to respond to the following questions. So how does photosynthetic efficiency change over time in response to thermal stress? And do these changes vary across different coral species? Do color channels derived from standardized photographs of Multipora hispida exhibit significant directional changes during thermal stress? And is there a relationship between color parameters and photosynthetic efficiency? And what does this reveal about the connection between visual appearance and symbiont health? So our study site was Morea, which is part of the Society Islands in French Polynesia, and has been recording bleaching since 1994. The sampling was done between 0.5 and 1.5 meters depth on the southern shore of the island. And our experimental design was inspired and tried to follow as closely as possible the paper I mentioned before. So after the sampling, we had an acclimation period of a month, and then a temperature round. So the temperature was increased from 28.7 degrees ambient temperature to almost 31 degrees. And then it was maintained for the last 11 days when it reached the 31 degrees. And during this 32 thermal exposure, the fragments were pulled from the tanks daily, and they were measured with pump fluorometry and and then they were photographed. So we had a total of 10 flow-through experimental tanks, and then 600 coral fragments, 4 genera, and 10 species. So each coral was taken from each tank, and it was measured with pump fluorometry. And this is a non-invasive proxy for symbiont photosynthetic efficiency. And we had a 30 minutes dark acclimation first, and then 3 different replicates to account for spatial variability, and then a 15-minute interval between each replicate to prevent photoinhibition effects and saturation of the photosystem too. And after that, the fragments were photographed with a Canon PowerShot G16, and then the settings were set manually, and they were kept consistent throughout the whole experiment. And then for the species Montipora hispida only, we use Adobe Photoshop to extract color channels, so RGB, HSB, and CMYK. And then we run statistical analysis in R. We run one-way ANOVAs to assess temporal and time-based differences in environmental parameters, then linear regression and non-parametric tests to evaluate species-specific photophysiology, and linear regression for color analysis and correlation with PAM data. Moving on to our results, I'm going to talk about each single research question, starting with research question number 1. Here we have photosynthetic efficiency over time for all the different species we analyzed. We ran a Kruskal-Wallis test that was significant, so there was a significant difference between the species, the 10 different species. So we ran a post-hoc test after to establish a hierarchy between the coral genera, and we saw the Posilopora species were the ones that performed the best. They had resilience, probably due to the fact that they host clade D symbionts that are known for being thermally resistant. And then Posilophoras were followed by Acroporas and Montiporas. And the corals that performed the worst were the Porides. So this could be due to the fact that the tanks were in a flow-through system, so all sorts of organisms would enter the tanks, and there are some nudibranchs from the genus Facilla that are that predate specifically on Porides in the area, and even if I remove them daily from the tanks, this could have added an extra stressor on these corals. And now we move on to photosynthetic efficiency over time, but with all the species pulled together. We saw a significant negative trend. So there was a decline of 1.12 units per day. So photosynthetic efficiency decreased, and this is due to the progressive photosystem 2 damage and the symbiont loss. So thermal stress is found in literature to be decrease in photosystem 2 function, increased photoinhibition, and decreased rubisco, which is an enzyme that takes part into the Calvin cycle. And however, only 24.7% of the variance was explained. So there might be any other processes that take part into this observed phenomenon. And moving on, here we have a visual analysis of how the different coral genera responded over time. So we have on the top row the ambient temperature photographs. So we can see that the corals have bright colors and their typical colors, while in the bottom row we have the increase of temperature, and the corals were paler or even white, so they bleached over time. And to compare our results to the previous paper in Hawaii, we have this graph that shows same thing, so photosynthetic efficiency over time, and in orange we have our own experiment, and then in green we have the experiment done in Hawaii. So I retrieved the data from the previous paper, and we saw that overall corals in Moorea performed better than in Hawaii, and Posilopora species in Moorea are known for being thermally resistant, while Posilopora mandrina, which was the only Posilopora species that was analyzed in the Hawaii study, is known for being bleaching prone in Hawaii. So the 4 Pusillopora species that we analyzed in Morea might have been boosting our mean photosynthetic efficiency. However, the very remarkable thing about these results is that the Hawaii study was conducted during the cooler months, while the study in Morea was conducted during summer. And despite these harsher conditions, because the fragments were already exposed to higher temperatures, they showed a more gradual decline compared to Hawaii. So these findings reinforce the bleaching resilience that is well documented for Morea in literature. And now we focus more on Montipora hispida, which is the species that we chose for the color analysis. Same thing, photosynthetic efficiency over time. We see a decline for this species, and it's in accordance with the other species. So it's also in accordance to literature, because Montipora species were found in different studies to have similar declines in photosynthetic efficiency under heat stress due to the photosystem 2 dysfunction and photoinhibition processes. And we can now move to research question 2, which focuses on a color analysis. So first of all, we can see how over time the color of Montipora hispida became from a bright purple to a paler purple until becoming completely white. And to see this in a quantitative way, we can look at these graphs. We have color values over time, and I'm only showing the 2 graphs that had significant results. So I have red that increased over time, and red was found in a recent study to be the best RGB predictor of symptom density and chlorophyll. So it could be a good indicator of coral health. However, our R-squared was extremely low compared to the one found in the study, and in general it's not a good predictor according to our results. And then we have Ki that decreases significantly over time, and this could be due to the tissue lightening. So since Ki represents black, the corals could be lightening over time due to the thermal stress. And all the other color channels were not significant. Then we move on to research question 3, and we are analyzing the link between visual assessment and physiological response. So we have photosynthetic efficiency in the y-axis and the different color values and color channels on the x-axis. And we saw that saturation has a negative relationship with photosynthetic efficiency, and this could mean that highly saturated colors might indicate the presence of stress pigments, for example, a fluorescent protein like the GFP. And then yellow and magenta also had negative relationships with photosynthetic efficiency, and this could be due to pigment or symptom loss. And blue, at the contrary, had a positive relationship with photosynthetic efficiency. So this could mean that darker colors are linked to better photosynthetic efficiency. And all the other color channels were not significant. However, even the statistically significant results had very weak relationships, so we must combine color analysis with other metrics for a reliable health assessment. To explain this phenomenon, we can say that color is a mix of different pigments, and not all of them are photosynthetic. Some of them are photoprotective, and this could create a mismatch between color and photosynthetic health. To conclude, we can come back to our research questions. We saw that photosynthetic efficiency declined with stress, and there was a significant difference between the species analyzed. Color shifted subtly, and changes were weak but directional, and color poorly predicted photosynthetic efficiency. Even if this study contributed to the general body of knowledge, we encountered several limitations. For example, there was no control, and this prevented any statistical comparison, and the tanks were unstable because it was a flow-through system, and the experiment was pretty short. All the color analysis and extraction was done manually and on a single species, and there is some missing data. So for future perspective, it will be interesting to expand to more species, more islands, and a multi-season sampling. And automation of the color analysis would decrease bias, and also the color extraction was extremely time consuming, so it would help also with optimizing it. And then add some data that we extracted during the experiment, so symbiont density, chlorophyll, and protein content. And this would allow us to develop a practical regional specific coral monitoring tool. So our take-home message is that this thesis represented the first few steps to develop a site-specific coral health monitoring tool. We emphasized the need to combine visual and physiological indicators, and we confirmed the remarkable reef resilience of Moorea and contributed to making coral monitoring more accessible and engaging the community. Thank you for your attention.
Thank you so much, Alessia. So we have some questions for you.
Yes, a couple of questions on your technique, on your method, and then another comment. So I didn't get if you were measuring the photosynthetic efficiency in vivo without extracting the zooxanthella from the coral.
Yes.
Or you first extracted the symbiont.
No, that's correct. It was without extracting them, so we had the different coral fragments, and we would just measure them directly on the. So we had 3 different points on the fragment to, and we tried to not do the same measurements over and over on the same area, and but it was directly on the coral tissue.
Did you measure the density of the zooxanthella?
Yes, we have symbiont density, but I had not access on those data. Yeah, it's for the paper, so yeah.
So I was just thinking, since you said that your samples were more resilient than the same species found in Hawaii, do you think that the fact you performed the experiments during the summer could mean anything in this resilience? I mean, do you think that the organism might acclimate to the summer and therefore show more resilience?
Yeah, it is possible. There are different species react differently to thermal stress, but there are several studies that try to first expose corals to a temperature stress, and then after some time put them in a bleaching state. And it's true that the results are not always in accordance with each other. So there are some corals that respond better after a previous thermal stress, and some of them don't show any difference, or they're already more stressed, already paler. But this could have been definitely a confounding factor.
Thank you, Alexander.
Yeah. So one thing you said that the results were not, you had low R-squared values, but I saw that the P values were significant. So what was
Yeah, I mean the results were significant, but let's say the predictive value of color was not. I mean, especially for these relationships, there's no clear predictive value, let's say, of color predicting photosynthetic efficiency. So that's what I meant. But yeah, we had some significant results, but compared to other studies, I found that the R-squared were pretty low.
I'm glad you bring this slide up, because I had a question about this. This is a very strange pattern. You have a band in the middle, and then you have what looks like outliers. Can you explain that? Why do you have all of them have the same, this line of points above and then 1 or 2 lines below? What's the explanation for that?
Yes, I think this could be a problem with the color extraction, because when I did the analysis, it was all manual, and sometimes there were a lot of shadows in the corals, because Montipora have I don't know, but it's like the shape of the coral creates several shadows. So it was difficult to erase the shadows and still have enough tissue to analyze and extract the color from. So I think that the outliers that I had are linked to overexposed overexposed area, so where the flash really made the coral wider, and then the shadows that made it darker. So there were some outliers in my data.
So, final question in terms of practical applications for monitoring. This work would involve like divers, I suppose.
Mm-hmm.
But do you think maybe perhaps in the future using ROVs with very high quality cameras could be used to map or take pictures of large areas?
Yeah, I think that it would be interesting to have ROVs or any kind of automation for to collect photographs and then maybe use AI or some machine learning tool that could use the color card and color reference card to detect bleaching stages automatically. And for the divers, yes, the idea is the color card is waterproof. I mean, the Morean color card is still not ready, but the Hawaiian one is waterproof, and it's meant to be used by even tourists that are snorkeling because the reefs are pretty shallow, and divers, and yeah, anyone could use it without a specific knowledge or training period. And for the Hawaiian color card, they had a validation period, so they had a prototype, and then they gave it to a number of students, bachelor students, and people that didn't know a lot about corals, and they asked them to do a health assessment, and then they validated the color reference card statistically and improved it to make it more accurate, and this will be done also for the Morean color card.
Thank you. Yes, thank you very much. Again, you was wondering, here you are looking at color band by color band effect on the performance of synthesis. Why didn't you try instead to look at the spectral signature, that is the shape of the FFV versus the different waveband? Consider that as a waveband. So there maybe you could see some spectral signature which change. So you didn't try to?
No, actually the project was mostly focusing on the color channels, so I didn't do it, but yeah, the main idea was to look at how the color channel would be linked to the physiology of the coral. But it's true that it would have been a good addition, especially because these results, as I said, they're not really, the R-squared are really low, and maybe it would have been interesting to look at the spectral.
So maybe, maybe I can try to collect the data. So this question 3 was only addressed for these species?
Yes, only Montecarlo speed.
Why did you choose this one and not the other?
So we chose this one because it had photosynthetic efficiency response that was kind of intermediate. So it wasn't the Poeciloporas that were extremely resilient, and then the varieties that responded really poorly. So we wanted an intermediate response for the first. The analysis will be done for all the different species, but this one was the one that we chose at first. And then it's also because the color of this coral is from purple to lighter purple to white during bleaching, so it was an easy and nice color gradient. So that is why we chose this, but the other species will also be analyzed. We only did it on one species because of time issues. Yeah, the color extraction takes a long time.
Then you propose to use other metrics than this one?
Yes.
Are you thinking of metrics that can be used widely in citizen science, or are you thinking of metrics that could be in the hands of the researchers, because the tools are mainly in the hands of the researchers?
So to make it more accessible to everyone, what I meant is that we should analyze also the other data that we extracted, so the symptom density and chlorophyll and protein content, and relate it to colors so that we can still make a color reference card for anyone, for all the stakeholders. And however, if there is no relationship also with those data, then it would be difficult to engage the community. But generally speaking, I think there's going to be a relationship with one of these metrics that these data that we collected, because if the coral is stressed, then it bleaches, so...
It's strange because you see by eyes, but you don't see it in your photo.
Yeah.
That's really strange. And I've been thinking also that this was a methodology to be applied by citizen science, that the environmental light is also very important in the photo which is made. And the photo quality depends also on the photo on the camera.
Yes, for the lighting, we always white balanced every time we took the picture, and we made sure that the lighting was the same. And for example, one day there was no power in Morea basically, but since we white balanced, we always could correct for these light changes. So the pictures were all corrected for this, and then in the field, people are going to look at the color card that is already validated, and then the actual color, and of course with different lightings at dawn or during the day. Yeah, this is something that we have to consider when the citizen scientists will collect the data. Of course, it's something that will have to be taken into consideration when analyzing the data provided.
Thank you so much. Any questions from, we have 3 minutes, so any questions from you? I heard you are not. No? Okay.
Can you see the features of the corals? Like then before and after bleaching?
Oh yeah. This one or the one with all the... Yes.
I mean, I've never been to Korea, but they look quite pale to me, even in the initial stages. So maybe like your methods are very good at doing power trend, maybe your resistance stronger because these are already starting to be stressed. Like maybe you've been lasting simple for a longer period, and because I know you couldn't get there for the summer, but maybe you could have been there in October, then you would see the full decline because they look, I don't know, they look quite pale to me. But
Yeah, no, honestly, they could have been already stressed compared to what they would have been in the field. When I arrived, the cores were already fragmented and in the tanks. So I didn't see how they were in the field initially, but the temperatures were already extremely high. They were more than 28 degrees Celsius, so they might have been already stressed.
I think maybe just like the end of the trend, which is why the like values aren't as clear as you think, but maybe like the trend is actually very strong when you saw the tail.
Yeah, no, this is possible. That's why it would be interesting to do a study with different seasons, because just doing it in summer, and maybe there's also another sort of stressor that we didn't consider, any confounding factors, but yes, sampling also during winter, for example, would have been very interesting, and comparing the two results, yes.
Okay, that's it. It's done. Congrats. Okay, and the last one is Elounda. Okay, last but not least, you are ready?
Yeah.
Go ahead. It's your time.
Welcome to my talk, Slugs in the Spotlight. My name is Hannah Douglas, and my project was focusing on the role of Kleptoplasty in Southern European Elounda species. My supervisors were Dr. Maria Rosalia Martin-Hinvas and Dr. Patrick Krug. So in my presentation, I'll give a brief overview of what is kleptoplasty, why is it important to study, the specific objectives of my study, my sampling sites, focus species, brief mention about my methodology, and I'll go through some of my results. I'm going to go through Just a few highlights of my results due to time constraints, definitely not all of them, but I'll talk about a couple of the interesting ones and then I'll conclude. So, what is kleptoplasty? It might be a similar sounding word, kleptomaniac, comes from the Greek kleptes, which means thief. It's a biological process in which these saproglossin or sap-sucking sea slugs sequester or steal algal chloroplasts from their prey species, and they sequester them and they become known as kleptoplasts once they're inside the animal, and they continue photosynthesizing within the animal. And the idea is that they can confer autotrophic or photosynthetic benefits to the slug. So why is this important to study? Well, besides being something that sounds like it comes from science fiction, a really fascinating adaptation in my opinion, it also gives us interesting insights into the early endosymbiosis that gave rise to the first chloroplast itself that revolutionized eukaryotic life. So this is kind of living analog for this process, and it gives us this kind of insight into bridging the gap between plant and animal biology. Additionally, it's important to study because although it's well documented in literature, the ecological and physiological roles of kleptoplasty are still quite debated. So it's not known necessarily whether it's a starvation adaptation for when the slugs don't have food to eat, or whether it benefits them when they're able to eat as well. Additionally, most previous studies focus on just one species or one metric, so I'm wanting to fill in that knowledge gap and look at multiple metrics. So my first objective was to look at the effect of light and food on a variety of fitness metrics, so looking at growth in terms of weight and length, fecundity in terms of egg laying, and feeding rate in terms of how much algae the slugs are eating, and then their photosynthetic efficiency. Additionally, I wanted to look at an algal comparison, so looking at where the source of the kleptoplast or chloroplasts are coming from, and whether this impacts the kleptoplastic functionality or benefit. And then lastly, I wanted to look at 3 novel Elounda species that have never been studied before and see if they have kleptoplastic abilities, whether they're able to retain chloroplasts as with other in the genus. So my sampling sites were in Nice in France, in Macron in the south of Spain, and in Lanzarote in the Canary Islands. These were my focus species, so I had Elounda virilis coming from Nice, Elounda timida coming from the south of Spain, and Elounda CF subernata, Manrique, and Levis coming from Lanzarote. Those were the new species. Just a quick note on CF subernata. Elounda subernata is a Caribbean species, and it's being thought that the same species exists in the Canary Islands, but very recent phylogenetic analysis has showed that it's actually a separate species, and it's in the process of being renamed, so we're calling it CF subernata for now. So my methodology, very simple, straightforward, 2x2 factorial design. I had 4 treatment groups, and this was in order to disentangle the effects of light and food, so that I'm able to look at them together, their effect together, but also separately. So I had slugs fed with ecologically relevant algae in the light and food and the dark and food, and then I had slugs that were under starvation conditions in light no food and dark no food. So to assess the fitness metrics, again quite straightforward, I used ImageJ to measure their lengths, precision violence to weigh the slugs' body mass and the mass of the algae that they were consuming and their egg mass, and then used pulse amplitude modulated fluorometry or panfluorometry to measure their photosynthetic efficiency of the 3 novel species. So a quick run through my results. I'll look first at the fitness assessment, the algal comparison. I'll give a sort of overview of what that looked like, and then the photosynthetic efficiency of the novel species. So starting with fitness assessment, this photo is probably my favorite picture for my thesis. It is the same slug. It is a slug that experienced a 3,700% increase in body mass in 24 days. So it's a very happy slug. So looking at the actual results, these are both graphs of Elounda viridis feeding on 2 different algae. So this is feeding on Pedomorpha, feeding on Bryopsis,
and these are two different species. So these are kind of the synopsis of the results I'm going to show you. This is looking at growth and weight retention. So this is the daily percentage weight change of the slugs. In pink we have fed, and in blue we have unfed. So across the board there is significance. If they're fed, they grow more, they retain weight better, and that's kind of what you'd expect. There was an overall trend of an additive effect of light and food, so when they had light, they seemed to also grow more or retain weight better, and this was significant in Timida and Levis. And then looking at feeding rate, so this is the change in, these are the same species, and this is the change in percentage algal weight. So if you see a greater decrease of algal weight, it means they were feeding more. And in Elysia viridis, on the two algae that we have here, it was clear that they were essentially feeding more in the dark. This is really interesting because previously we saw they were growing more in the light, yet they had to feed more in the dark. With Timida and Levis, we didn't see this effect, and it was more subtle. And I think there's a variety of reasons for that I can get into later. So fecundity, this is total egg mass that the slugs were laying. Again, similar to weight and length, the slugs that ate more laid more eggs. But again, there was this sort of observable trend, although not significant, that slugs with light and food, there was this additive effect that they laid more eggs when they had light. So briefly looking at the algal comparison. So this, the comparison was 3 separate experiments of Elysia viridis fed with 3 different algae. So the slugs gained more mass and length when they were fed with this algae, Ketomorpha, as compared with fed with Bryopsis. But when they were fed with Bryopsis, they laid much more eggs than when they were fed with Ketomorpha. So they both kind of benefited, just in kind of different and interesting ways. Those fed with Codium fragile rejected the Codium. So despite it kind of being mentioned in literature that Elysia viridis loves Codium, we kind of expected them to really like it, but they didn't. So I think the sea slugs from Nice that we gathered potentially have a local dietary specialization. If they grew up eating not Codium, they're less likely to switch onto it. Or potentially it could be a signifier of cryptid speciation, because it has been shown in the past that this genus, their algal host is a driver of speciation in this genus. So looking at the Kleptoplasty retention of the 3 novel species, we have Elysia manricae, Elysia subvernata, CF subvernata, and Elysia levis. So these were my PAM results for these 3 species. Overall, there was clear Kleptoplastic retention, so that was really cool, but we could see that all 3 species retained some sort of photosynthetic ability throughout the experiment. In manricae and CF subvernata, there wasn't a distinct difference between whether they were fed or unfed, and we were pretty sure that this would be a problem with the algae that they were given, with the Kleptoplasty webiana. We weren't able to grow the Kleptoplasty in the lab, and we think that maybe it could be because of this, maybe due to disintegration after it was taken out of the sea, and various other reasons, or maybe they just don't eat that algae because they haven't been studied before. But overall, we were able to see that even in the ones that were unfed, they continued being able to retain photosynthetic efficiency. The result here that I was really excited about and found really interesting was in Elysia levis. So you can see here this dashed blue line is dark unfed. So this is essentially the average of this group of sea slugs that wasn't fed. So for 25 days, not being able to replenish their chloroplasts, they still managed to maintain an FPFM photosynthetic efficiency reading of above 0.8. And for reference, 0.8 is around what a healthy plant would give. So this is an animal that has been starved for almost a month that is still continuing to photosynthesize at that level. It's important to note the difference between the light and dark. So all of the dashed lines are the slugs that were kept under dark conditions. And this is because of they have a higher photosynthetic efficiency because they're not
experiencing photo damage, so they're able to retain efficiency better. So a couple of limitations and future directions. I think, as happens very often, it would be great to have had more replicates. For some of the species, you could observe subtle differences in the data, but it wasn't statistically significant, so I think with more replication, I'd be able to get more of a clear answer. Additionally, it would be great to look more into the diet of Manrique and CF subordinata, because it said that they eat Caulerpa, but we didn't see that they were definitely eating it or not, so it would be really interesting to look more into that. And additionally, I think this kind of study would really benefit to be linked with molecular analysis, so to kind of add this behavioural physiological analysis with molecular to get a better understanding of kind of the underlying mechanism of plastic retention. So to conclude, my study showed that Kleptoplasty provides species-specific advantages, so the advantages varied between all of the 5 different species I looked at, and these varied between enhanced growth, weight maintenance, increased fecundity, reduced feeding effort. We considered reduced feeding effort to be an advantage or a benefit, because if the slugs have to spend less time foraging, less time feeding, they're less time out in the open, potentially at risk of predation. They also have more time to look for a mate, and also a lot of these algae are chemically defended, so needing to ingest less of them is potentially a benefit. And as I said, Kleptoplasty is context dependent, clearly influenced by diet, and the light regime and the physiology of the slugs. So some of the slugs we studied didn't have lateral parapodia, so these flaps that they can protect their kleptoplasts with, and you could see differences in that. And additionally, I said about wanting more replicates to get statistical significance, but I did have statistical significance in terms of seeing an additive effect of light and food on at least 3 of the 5 species that I studied. And I think that this could show that Kleptoplasty potentially enhances heterotrophy, so it aids in their feeding rather than being solely a starvation buffer. And potentially it could even be a co-limiting trait, which is normally something that you look at in an autotrophic organism, but potentially these animals are kind of blurring that boundary between heterotrophic and autotrophic, so potentially light is a co-limiting factor. And finally, my study gave the first empirical evidence of confirming functional kleptoplasty in the 3 species from Lanzarote. So I'd just like to thank my amazing supervisors, particularly Chari, who took me on board after I was supposed to go to Los Angeles and there were wildfires, and very last minute change of plan. She took me on board, and it was amazing. And Pat, who from afar was very helpful and always willing to answer our questions despite being evacuated from his home, and everyone that helped me in the lab and the field, and to the Jordan Moore Scholarship Programme that funded my Masters and vicariously this study, and to my host universities and the University Programme for the opportunity.
Well done, Karim, you are the person now.
Thank you for a very nice talk. You mentioned in the end the question of replicates, the need for more replicates, but you don't think you said in the beginning how many replicates you actually had, just out of curiosity, how many replicates?
I had 5 in each treatment, so for every one of my 7 experiments I had 20 slugs under the treatment, so yeah, 5 in each treatment.
I was just wondering, I don't know anything about sea slugs, but what's the lifespan of these? They live more than a year?
They can vary. Yeah, they can live about up to a year, but it can vary massively depending on the species. But yeah, around about they can have a full 12-month cycle.
So could this Kleptoplasty be an evolutionary advantage if there is seasonal variation in availability of algae, for example?
Yeah.
Yeah, massively. Yeah, it's a really interesting question. For Lysia specifically, this algae in a specific area experiences calcification during the summer. So it basically becomes inedible and they can't eat it. And Timida is known as a long-term retenting species. So they're actually one of the best adapted to be able to retain the chloroplast that they've gotten during the other seasons. So yeah, very much so it could be a seasonal adaptation.
Thank you.
Yes, thank you. One question about the method. In the dark grid, it was fully, I did not get it, was it fully dark or it was?
It was almost fully dark, so it wasn't in complete darkness. So what we used was black tights material on the top of the containers, and they were in the same space, so they were under the same temperature and same water and everything as all of the other slugs. But we used a photometer to test the light that was coming through at the level that the slugs were at. And if I remember correctly, it was between 10 and 14, and I haven't got it written down. I think it was between 0.10 and 0.1.
So, which means that the photosynthesis of the microalgae was stopped?
Yeah, that's correct. Yeah, well, the thing was that we kept, so I had all of the different algae in different aquaria separated from as in the stock algae that we were growing. And depending on how much they were eating, I was kind of continuing to replenish. So they all had about 0.1 of a gram of algae in their containers with them. So some of them, so for example, Kleptoplasty or Bryopsis, the Viridis were like voracious and they'd eat all of it within like 3 days and then they'd replenish it. So it was.
Okay, so I did not understand that you were replenishing with this food, I mean with the new algae. The PAM measure, I'm not familiar really with it, does only provide information about the efficiency of the statistics, also on the quantity of carbon which is fixed.
It doesn't. It can be used as a kind of proxy for carbon fixation, but what it's actually giving you is the photosynthetic efficiency. I think I have a slide for it. So it essentially measures. It measures both the baseline, so the minimum fluorescence that it can give back, so the minimum amount of light that the chloroplast can absorb, but then also the maximum, and then the machine itself does the calculation and gives you the maximum potential fluorescence, and it can be used as a proxy for carbon fixation. But
at some point, it would be interesting to do a carbon budget of the animal.
Yeah, for sure. It has been done in other studies, and it would linking it with the kind of physiological analysis would be really interesting.
But in those previous studies, you just mentioned, what is the amount in total respiration of the animal? How much is related to the production by the algae? And how much on the other way of feeding?
Sure, I'm not sure if that's been tested. I'm not sure if they've looked at the difference in the carbon budget of like what they're eating and the carbon that they're actually consuming and what's being created via photosynthesis. I'm not sure if that's been looked into or not. I know, actually, yeah, I'm not sure. I don't want to say something wrong.
Thank you, Lord. Any questions from the public? We have 2 minutes. Yes?
I just
I'm
have
sorry.
Sorry.
you said that they are feeding during the night and growing during the day. Did you say that? Or sort of?
They feed kind of all the time. It depends on the species. So the species from Lanzarote, I actually sampling for them, I found them pretty much always just at nighttime after sunset, because that seems to be when they kind of come out and about. But I'm not sure if that's because that's when they're feeding, or if that's just when they come out because there's maybe less sunlight, so they're less likely to experience photo damage. But they'll eat all the time. Yeah.
And is there a literature saying that it can also feed on microalgae or
on macroalgae?
Micro.
Oh, micro. I'm not sure actually. That's a good question. As in they can eat it off like epiphytically and stuff like that. Potentially, I'm not sure. I don't know of any species. I haven't found any species in the literature that eats microalgae. Most of them are saproplosin or sap-sucking, so they pierce the algal cell and sort of suck out the contents within the thallus, but yeah, I don't know of anything.
And last curiosity, what is the FB or FM value of empty sea slugs?
Of empty dead sea slugs. It actually it gets lower and lower, so you can get a living sea slug that's kind of turned totally white and doesn't have any chloroplast. It might be around like 0.2 or something like that, but it actually, the PAM stops being able to read it and stops being able to pick it up when they kind of go past. So I think if there's even a tiny bit left, it will pick up kind of 0.2 is the lowest, and after that it will just give an error signal. So yeah, and some of them I'd have like a slug that was still alive, but it would give back an error. So.
Thank you, Hannah, just a doubt. In the phylogenetic from related species, there is a moment in which Kleptoplasty appears, and that means something. I mean, because in the related species, how many of them use that as a strategy?
Most of them do. It hasn't been studied, but I would imagine that the majority of Elysia species would. Kleptoplasty actually evolved twice separately, and not within Elysia, but within Thesis, I'm sure. So yeah, I'm not sure. There's a lot of sea slugs that aren't able don't have kleptoplastic capabilities. But within Elysia, I think a lot of them do. There's 157 known Elysia species currently, and not all of them have been studied. So, yeah, I'm not sure how many are able to do it or not.
Yeah, just a curiosity, because you had your 3 sampling sites that were very far apart. Is that because those are the sites where the species are specific? And also, your novel species, are they from the same sampling sites, or are they from different sites?
Yeah, so these sea slugs are, I would want to say everywhere, but they're not uncommon. All of them aren't uncommon. Like, for example, I'm not going to go back, never mind. Elysia viridis, sorry, I'll go back to the slide, okay. Elysia viridis is quite common. So, for example, I found it in Ancona here. The sampling sites were selected just for kind of ease of the study. Nice was where we were, and then Matheron was handy because somebody's parents' apartment was there and things like that, so it was kind of for ease of sampling. The 3 species from Lanzarote were all from very close to each other. They're near Arrecife in Lanzarote, and the CF Sobranata and Manrique were from the same kind of bay area, and then Levis was a bay very close by, but they all kind of, I think that was just where I managed to find them, but they're all over the place.
I wanted to ask whether the body size of these slugs have an effect, maybe for the split, actually they have I don't know whether you explored that option, because for example, the smallest slug system that you showed was the one that managed to have the capacity to photosynthesize more than to retain its capacity, So I don't know whether you saw
a difference between size and photosynthesis. Yeah, I'm not sure. The thing I know is in terms of measuring their photosynthetic capabilities, the smaller they are, the kind of trickier it is, because they're tiny, so you can only put the sensor in so many places on their backs. But I didn't see any difference between their size and how well they were able to photosynthesize, except for just the kind of fact that the Elysia levis were the smallest and they did really well.
Thank you.
Last question.
I'm wondering, since you collected these from the field, I'm assuming they weren't all the same size and weight, do you have to account for that somehow?
Yeah, that's a really good question. So I actually, 3 different things to try and account for that. So in terms of the methodology, I put the different sites. So I looked at all of the sea slugs, did an initial measurement of all of one species, and then I randomly assorted using a random number generator all of the different sizes to all of the different treatment groups to make sure that there was some small and some big ones in each treatment. And then going through the sort of data cleaning process, how I got the data for weight or length and fecundity was I basically took away the initial weight, so I got only the percentage weight change, and then from that I did an analysis of covariance using initial body weight as the covariant, so kind of statistically to offset for the potential of the difference of initial weight affecting the stats.
Okay, Hannah. It's done. Thank you all. Grazie a voi delle studenti.